as you know, Lake Conference happens once in 2 years, and it started initially in 1988, and ever since, I think from 2000, KKEHS school has been a part of Lake Conference since 2000.
1998.
1998, sir.
Yes, first one.
Okay, so right from the year one, this school has been taking part in the conference, and we have seen wonderful projects from here. Okay, so this area is quite unique because it's not completely urbanized yet. Okay, there are some patches of green areas around, there are good lakes around. Okay, there is hope to save the natural resources that we need for our survival. Okay, whatever we say and read, saying save animals, save plants, we need to. But why we need to? For our own survival. Right? Of course, they have the right to live, but they don't need us to, go feed them or take care of them and protect them and all those. They will do that on their own. But why we need to protect those habitats is mainly because we need it for our own survival. Because there are a lot of ecological services that we enjoy if they exist in the natural habitat. Okay? So on that note, I welcome you again to the 4th Three Lakes Orientation Program of Lake 2024. Okay, so today we have a series of sessions planned. I hope you have seen the agenda. Due to unavoidable circumstances, TVR sir could not come here and present the lecture directly. Instead, he has shared the video with us, which we will present later. So we will be, streaming his video for you all. Please listen carefully and write down the notes. Later on, he will be joining us. So then you can have an interactive session with him where you can ask the questions to him, and he will be more than happy to answer all your questions. Is that clear? Okay. So all of you have a pen and book?
Yes, sir.
And your mind is fresh and empty?
Yes, sir.
Emptier. Empty as in you have a lot of space to accumulate a lot of information, because we have a series of sessions for you and we will be giving a lot of information to you. More than what they are, we will be just touching upon the basics and then teaching you more on how to document, how to collect data. Okay? Because after this program you will start your projects, and for those projects you need to know how to collect the data. Once you collect the data, your teachers will help you analyze that data and make it into a lovely project. Okay? And then you will have to come and present your project in the month of July in the school itself, and in August, maybe here or someplace else, you will be called to present your projects, the selected people. So from July onwards, the selection process will begin. Okay, so if your project is good, you will come to the next round, that is in August, you will have another round of selection. And if you are selected in that round, you will come to Mudbidre in October, which is close by to Bangalore city. So you will come there and you will present your project in the conference. Okay? And the conference is an international event where people from across the globe will be taking part, and you will get an opportunity to present your project in front of all of them, and you will get to listen to projects across various areas, different habitats, different geographic regions, and learn from them. Right? It's a great opportunity for all of you at this age to have such a big platform to explore and learn. Okay? So I wish you all the best for learning today and also for your projects.
If you pollute the water body, that means when you pollute the water body, there will be scarcity of water, and there will be fight for clean water, this time. That's why how do they practice? If you visit Dargah, you have to wash your feet and finger with the clean water. Then only you can go to Dargah. They thought that when you practice in Dargah, you will also practice at home. Okay? Now look at another scripture. For example, Masya Purana talks about that. Masya is fish, okay? If you have a well at home, go well eat. Okay, 10 wells are equal to 1 day, 10 lakes are equal to 1 Sarovar, 10 Sarovar is equal to 1 child, and 10 children are equal to 1 tree. Okay? Now, I put it another way. When you have a tree, you are nurturing your own 10 children. When you remove water body, you are nurturing your own 1,000 children. See how effectively our elders have communicated to us. Why we should respect Mother the way your mother works. When children respect mother at home, that home they will get prosperity. When they respect mother at home, they will also, as they grow, they respect mother. When they start respecting mother, there won't be a quality of mother. As you all know, we have only one mother. That's where we have to realize the degradation happening in the system. This year, the World Environment Day, we celebrate once every year on 5th of June. And 22nd May, we celebrated Biodiversity Day. Now, what is the theme of the World Environment Day? If you look at the theme, it is a land restoration. Okay? And then revitalizing the ecosystem so that you are not resilient. We are talking about drought. How to be resilient? When you have water bodies in your locality, when you have greenery in your locality, when you eat food which are nature friendly, then you are strengthening the system which is not resilient. Let's see whether the connection. So when you do the practice of your life, you are achieving this. And as a student of science, I will request you to do analysis of the water, which is very important. Okay? In this form, we will sometimes see the traces of dioxin in this form. In the traces of dioxin is a constant which is a resistance. So let us not give this to anyone who we know who will be respect and give them the answer. Okay, sometimes we get to happen of ignorance and it is an opportunity for us to educate this kind of things. Okay. So now, one message to all of you is to not to use plastic as much as possible. People are people know the whole thing without thinking they are being carried. They think they are too easy people. And they get hooked in the plastic container and quality impact. Just analyze the food content. You will be surprised again to see the chemicals in that food. That means we are doing our family members because of our average of not taking the container. We are giving the answer. Let us not do that mistake. Anyway, this kind of is help you understand this kind of the essence of the subject and the behaviors you understand. So the purpose of this symposium, we have been doing since 1998. This is a 14th year. There are about 1,000 to 1,200 participants recorded in the event. There will be international experts, national experts, and the decision makers. Apart from that, 10 to 15% are students from schools and colleges. Then I could have conducted the event without school children, which is also easy for me to do. But I always believe if I give an opportunity for the school children to take part in this kind of international forum, when the child attends the symposium, how the 3 days, the child will get the knowledge. Then, on the concluding day, when the child does the presentation of this or her work, when they get appreciation from the audience or expert, the confidence level goes. That's what we are looking for. And our agenda is to make every child to be confident so that they can become highly successful in their career. Whenever we talk to the children, we ask them, what do you want to be? Immediately I get answers saying that I want to be a doctor, I want to be an engineer, like that. First, they say, let us be humans. Let us think of our neighborhood. Let us think of our children and their parents. and then think of what you want to do. Once you start respecting your mother, you will sometimes see you will be highly successful in your career. You can do anything. You can become an engineer, you can become a doctor, but you will be able to do it. If you miss that, there is no point in becoming an engineer or a doctor. See, today, I have seen doctors without humanity. The profession has become so much. They want to just make money. Money will not be everything. When you take a sick person to the hospital, if the doctor demands 5 lakh, 10 lakh, you can imagine how the situation is. So that's why for the doctor to be a human being, good human being, let us give the essence of environment. Environmental education makes the people sensible. When they become sensible, they will be responsible. That's what our idea is. And we are successful. Many of my students who are participating in coaching, today they are faculty in various leading institutions, including NASA. So that is what we are looking for. And respect the children's start thinking. When you start looking at the environmental subject or the nature, it's an interdisciplinary. You can look at the environment as an engineer, you can look at the environment as the science student, you can look at the environment as a doctor. You will understand as a progress, I will illustrate all these things. Okay? So now the World Environment Day, the current year theme is to restore the degraded land and also make the system prosperous. You will see a photograph on the screen. Can you tell me what is this? We should be proud of our heritage. This is the family tree, a huge family tree in West Bengal. We saw almost 140 acres. I was told by a student that the main plant has been removed because of the fungus attack. Now the family tree has spread over the entire village. It is like parents leaving the sea and children shining in the system. Exactly a good example for the Indian families. Indian families believes in joint system, joint families. We want our grandparents to be with us. We want our parents to be with us. We never wanted to be alone. We were never selfish. But unfortunately, the education, what we have teaching in some of the schools and colleges, have made the children more selfish. That's where they grow, they never think of their parents nor their family. I hope those things will change with time. Now, one is when you see something is degraded, if you restore that ecosystem, you have done your job. You might say that, I'm a child, how do you expect me to restore? See, if you do not pollute the system, when you eat something, if you do not litter it and dispose it scientifically, you will have done your job. When you look at the plants, if you allow the plant to nurse, you have done your job. When you look at the water bottle, just go to the water bottle and meditate. You will see the kind of satisfaction you get. Okay? So, now, today I am talking about, I will explain about your ecosystem. Know your ecosystem. See, your parents will be talking about know your customer in the bank. When you have a bank account, you have to provide some document. The bank insists for the document so that they have KKEHS. Something similar. You need to have know your ecosystem. This is told by one of the people conducting the World Wetland Day there. In Chandigarh. She was just a spectator. She saw our program. Then she told me, sir, your actual know your ecosystem seems to be very catchy. Because this is what everyone should know about. The moment we know about the ecosystem, we will know how to manage it. Okay? So, now, today we have thought about the program like this, okay? After my presentation, Viju will talk about birds. Then Yurika will talk about how to do the mapping of the spatial mapping. Then Gadana will talk about the water quality and soil. And then we are at a day you should go back to your neighbors, you should also educate them. You should also go to the other classes and explain the same thing what we have explained to you all, so that make every child in your system knowledgeable. Okay? Now, this is the web page where most of our research articles are posted. That is www.IISC.ac.in. In this page, there is the lake symposium page. If you are interested in another thing, you can go through it. See, we have created a Bangalore Urban Information System, Bangalore Lake Information System, which Abhishek is going to talk about it when we talk about artificial intelligence. Okay? Then the lake symposium, when you access the web page, it gives the details about lake symposium, and more importantly, we have students and teachers around. You know, here we have to put the reference material, how to do the study, how to document it, what is the format to be followed, and also the video section. You know, here we are going to put, we have put all the videos by, you know, the top
Yes,
Sudarshan, all the Cincy, all those, you know, Manna lecture we have put there so that you can access that anytime. Then, as part of the lake symposium, we also conduct a orientation program in various schools. You know, naturally, when I go to the schools, most of the schools don't have the environment education, environment science teacher. You know, then we thought that we need to conduct the orientation program. That's why we conduct the orientation program. Now, this is the third event what we are doing in the city. First, we started at Punakumani School and Anantana. Then we moved to the Vidyanagari, we are in America. Then today we are here. And then next week we are going to the Varthur and Varthur as well as KKEHS school. Then also Vidyanagari and we have a network of, do not think that we do only this. Okay? We do science. We also take classes for the students. In addition to that, we do this program. We don't go to hotel and or a restaurant on Saturday afternoon or Sunday afternoon to enjoy the food. We go to school to enjoy our life in this age. That's the benefit we do. Okay? So now I will request you to go through this web page wherein you get all the details. Okay? And this video also we are going to put it online. Okay? So now the process is not like this. We conduct a orientation program in the school. Then students and teachers will carry out a field-based day. That is, immediately after the orientation program, we will start it. Then there is an evaluation by the respective school. Then they shortlist the students and they will send a list to us. Then we will come and take the final we conference wherein this selected student we will evaluate once again, and then among that, some of students will select and take them to the main conference. But in case if someone is not selected, don't be disheartened. As part of the lake symposium, since you have taken part in the orientation program, you have an open invitation to attend the lake symposium. You can come to our, we will arrange your accommodation, we will provide you the food also, so you are most welcome to come. See, when you come there, you can listen to your friends, you can listen to the experts, etc. Okay, that's what is going to happen in day 2024, that is between 17 to 19 October. Then on the 20th October, we will go to the nearby area, the field visit, either the second road or the lake in the vicinity. Okay? Then on 20th October, late evening, we will plan to return to the Bangalore, directly for Bangalore, so that I can take up my other assignment on 21st October at IISC. Okay? I officially invite you to the lake symposium. Okay? So, you are all invited. So, you are our guests, you are our children. So, you will learn with us. Okay? And we will also learn through you. So, you might wonder why I have made this statement. But learning never ends in our child. When I go to some school, when children get a new idea, we get excited. We want that to happen. And I'm sure as youngsters of today, you will try to do that. Okay? on the terrestrial ecosystem and algae in the earth body, they essentially fix that energy during photosynthesis. They convert the carbon dioxide which is in the atmosphere into the oxygen and also the food. That is the food that is available to the next level. Had the plants that used to provide food, no one will survive on the earth. That is the importance of the plants. Okay? Then the hydrophobes, they help to transfer the energy from one level to another level. Then finally you have the ability which remove the regret, the items or the walls, they prevent whatever is left in the system. Okay? And what they do, they convert that organic fraction into electrical form. Okay? Then we have aerobic component that is essentially surrounding us. The physical and chemical aspect, the physical, the solar radiation, temperature, rainfall, soil, water, air, essentially what surrounds us is the environment. Okay? Now, let us learn the food chain effectively. As we said earlier, see the producer produces food which is eaten by the next level, herbivore and carnivore. And finally, whatever is left in the system, they convert it into elemental form by bacteria and fungi. Okay? Now, when you see this carefully, you see that energy and material transfer happen from one level to the other. Why do you eat food? Why do you take food? Your body requires energy. Your body requires nutrients. So that's where you take the food. And when you take food, there is energy and material transfer happens in our body. Similarly, happens at various levels. Now, when you look at this carefully, there is nothing called waste in the system. Where do you see waste? You will see waste where waste people are. You know who are these waste people? The people who are accumulating plastic, you can put food at home. But instead of that, sitting at home placing an order for food through the Swiggy as a matter, are the waste people. Because they are too busy, but cooking hardly takes about that, the 45 minutes. How many 45 minutes? Most of us, even in IISC, we don't know how much food we cook. We enjoy the husband and wife, we join together, we cook, children enjoy food. But when you take the food to the city, what else? You do not know whether restaurant food is given to you. And in that plastic container, what all things you get into the things. See, there are all kinds of worry in the food. But I am worried about the chemical contamination in the food. That is where, and people say, the children who are sitting there, gentlemen, I want to say, when you are listening, you will shine in your way. Discipline comes in a hard way. And people like us, like me, know either the time or the resources. Okay. Now, when you have to control rodents, in a pair of rodents, they are offering in a year is about 400 to 500. In a 2 years, it's 200 million. Now, each pair will have 400, 500, you can imagine 400 into 400. How many of them? If you have a snake or a home, they control this rats or the rodents. But unfortunately, you see that today's adults, since they do not have the knowledge about snake, any crawling creature, they treat them. And they go and kill the innocent who are doing the great job to all of us. See, these two fellows who are having this kind of thing called animal. You should tell them what the importance of the snakes in our system. Okay? Because I was told in previous also the snakes keep coming to the classroom to listen to the teachers. So we should allow the snakes to come and enjoy the beauty of class in the classroom. We should not tell the snakes to enter the classroom. Okay? It's also indicated that in the classroom there are rats. That's why they have come. Otherwise they would come. That means if a teacher is feeling that a snake has come into the classroom, she has to make sure, he has to make sure that no rats are there in the room or in the campus. Okay? Now, we call this as a keystone species. Okay? And these are all the beauty, of the every component being a part of an ecosystem, eat and being eaten. Okay? Dynamic and complex interaction matters. And increase your understanding and knowledge of the ecosystem at the moment. You can take up any of these organisms and you can try to study. Look at their behavior, the function they do in the ecosystem. That would be your one of your scientific project. Don't tell me, sir, why should I study the approach? Look at what the approach does, then become resiliency. What is there in the system? Now look at why the rats are there. Okay? Or the snakes are there. Okay? I have talked about the environment surrounding us, I have done very in the detail. Now you see that we have used the technology to understand this concept so well. For example, simple water cycle. I take classes for the people on the lady teachers. The experts like you, teachers, and college faculty, etc. Whenever I ask them about water cycle, none of them have given me the answer which covers all these components: the precipitation, evapotranspiration, then condensation, cloud formation, then rain, and part of the water getting infiltrated. This is the most important part of what happens in the system. When it rains, the water gets infiltrated in the underground. That is the water in the groundwater. That is the water that you will get in your well after monsoon. So now you see that how the groundwater recharge happens in the system. I'm sure you can make up you can make up. And our study showed wherever vegetation is there, about 45 to 60% of the rainfall gets infiltrated. Okay? Now I will tell you one answer I had got when I was taking a class for the teachers from engineering colleges. They were about 60 years old. I asked them what is the water cycle. Today I will ask you because you are school children, I am sure you are teachers who are made sure that you all know and you have understood the concept. When I asked that one gentleman is water, I asked him what is water cycle. He told me, sir, you see outside. I thought it is raining outside. So he said, see outside, people are carrying water pot on bicycle. When you carry water on cycle, it becomes water cycle. Okay? See, this is the problem in the system. People, of course, we is a faculty, giving such a wrong knowledge to the youngster is the problem. That's why as a teacher, we have to be very cautious and listen about the next stage and understand the concept. That's where this kind of program will help. Okay? Now, you might ask me, what did I do with the diploma? I just walk out the.
Can someone explain or summarize till now what you have understood?
At least these two slides which have gone. You have seen some animation and all this, no? Yes. Yeah.
What's nature and how we can save it and approach how snakes help by killing rats and the water cycle. How it is used to filter the water that comes to the
This description you understood
Sir.
of the water cycle. In that, one important thing is explaining about how water seeps through. You would have studied about water cycle. The only thing is underground water recharge, which usually is forgotten, is also included here. Okay, which comes again later for us to use.
Say that I am free to take a class. Then of course some of the teachers came and said, just because the professor why you walk out. I said, no, I can come back. For the other people to take class on my own for the programming courses that I have. Then I traveled again after 50 days. Right? Carbon sequestration in algae, carbon sequestration in terrestrial plants, and carbon sequestration in the macrophage which are closer to the water body to see the soil in the wetland on the highest carbon sequestration. There is no highest amount of carbon. But to tell higher, you should do the experiment. You collect the water of the sample, soil sample, and the plant, you see that how much carbon is there in the carbon will be in the two form, organic and inorganic. Okay? So that is another experiment you can think about it, but it is also because in my subsequent presentation, we can do a presentation of the carbon sequestration. Okay? Now next.
You understood that slide? Who understood? No. What is carbon sequestration? How much carbon is correct, but inside water what's not removed? Inside the organisms, like different. You understood water cycle, right? Similarly, carbon, nitrogen, all these cycles you would have studied. So here in terms of carbon cycle, how much carbon is stored in a living organism's body? Okay, by checking, say for example, checking my weight, how much carbon is inside me can be assessed. Similarly, for a tree also you can calculate. How? By cutting the tree and weighing them. Should we do that? So what we do, there is a formula and there is a method of how to measure the height and girth of the tree. Okay? Taking that measurement, there is a formula in which you put that data, it will give you the biomass, above ground biomass, and then he will explain like how you can calculate the total biomass of the tree. Okay, so you can revisit this video and check that how it is done, and you can do that, and in case you have any questions, you can always get back to any of us. Is that understood? Why is that important? How much carbon it has harvested? Why it is important? Huh? Very good. So when we burn fuel wood, what happens? All that stored carbon is released back into the atmosphere.
Yes. Diversity you know that diversity can happen in three ecosystem, species, and gender. You look at the ecosystem level, the diversity. You know what is the beauty of each ecosystem? What is the function? Okay? Then similarly species. You know, for example, if you want to understand species diversity, take an example of banana. When you go to the shop, red is not banana. Red banana is there, Bengal banana is there for Kerala. Then we have our own varieties, Kerala banana and banana. But the chemical composition, biochemical composition of that, you will start to see each of these food, they are not just giving the food, they are also giving the ingredient because of the medicine product. You know, simple example is, if you take that Bengal banana, okay, of Kerala, which is rich in fiber, when you take the Bengal banana instead of the other banana, you will not have sugar control. Because the fiber is food, when you take the food, as the calculated system is slower, the sugar don't get fixed in your body. You will not have sugar. You know, that's where you when you go to the doctor, the advice you have food which is rich in fiber and also calcium. You know, even yesterday, the Indian experiment was in the newspaper. You have seen how the fiber rich and the protein rich food controls the thyroid. Today, in fact, the young girls with the age group of 20 to 40 or 60 having the thyroid problem. If you want to control that, the only way is to have the healthy diet. You know, that's why when you do this kind of experiment, you will understand the importance of diversity. You know, that makes you also, your family to be very happy family. You will start telling your parents, then your parents will buy item which is healthy. Otherwise, what will happen? Otherwise, what do they do? Morning to night they will eat only pizza. And they will come back pizza. You know, that is the priority, okay? So now, when you look at some of the species, they also tell us that.
Understood that part?
Yes.
say? Ecosystem, species and genetics. Very good. So what are those three types? What are those types of biodiversity studies that are done? Okay, so for example, that is showcased so that you understand. You can study what are the different species in your region, or in one particular species, what are the different varieties that are available. That becomes genetics. Right? What are the different genes? Then the entire ecosystem you can study. Okay, what are the different components of an ecosystem, how they interact with each other, and how you get benefited with that being healthy. All these things you can study and do for your project. Okay?
What else of the ecosystem? For example, if you look at this plant, it shows that only it has adapted to a situation where there is a high amount of moisture. You know, good precipitation has to happen. Now it has got adapted to that. Then if you do this job, but still it is not here. Similarly, the crops, you know, any changes in the environment, there will be some changes in the morphological features. You know, for example, if there are any changes in the surrounding environment, you also react actually. When you are exposed to a highly polluted system, you know, you will have tendency to contact each other and okay? And these are known as keystone species. You know, why do we call all as the keystone species? As I said earlier, you know, when all these binding tools are loaded, they are going to be as a fauna in the trade, conserving the trade. So the fauna don't use the similarly tigers, if you look at when tigers are there, it keeps control over the herbivore population. And because of the control of herbivore population, you know, your grassland will be maintained. Because the grassland is maintained, the population of water is. You know, that's how where tiger is there, you get water. If you don't have tiger, because of imbalance in the nature, then you will not have water. As it is happening the communication. See, when we don't have water in the community, what will happen? Two left fellows will start fighting. Okay? So now we understood what are keystone species. Then the next is the umbrella species. See, all.
Have you understood what are keystone species? What are keystone species? Yes. Help for farmers. Huh? Correct. Tiger, what it does? Whose population? Very good. They control the population of herbivores. Okay, so then what happens? Who controls population? Yeah, they play a major role in the ecosystem, because of which many things will be dependent on. Okay.
You should understand that and KKEHS are another problem. Okay, umbrella species because this was there. So many other species get. That's okay. Then you have the species which are beautiful. You should try to understand why they are beautiful. What makes this particular taxa to this kind of talent, and how it is useful for the human person. You know, they are coming from cultural point of view. Okay. They will be good people with that. So because of that, you know, the cultural significance you can understand. Okay. As I said earlier, those are the species of frogs. And there are economically important species like honey, what we get because of the plant nectar. Then you have some of the highly prized, you know, Ministica, all these. You know, this is what you get from the plant. Okay? And that's why you need to understand the ecosystem. Take your own world. Look at what kind of birds are there, what kind of animals are there, what kind of interaction happens. That is a good something. Okay? Don't try to do the copy paste. I was told there is something called ChatGPT. by only transfers. People who have real intelligence, they will never use the ChatGPT. When a school child using ChatGPT for something assignment to show the evolution of the species. If you want to be successful in your career, don't ever think of reading. Don't ever depend on Google for that. Go to your textbook and learn. Go to your teacher and take it back. Don't take Google under help and be around. See, Google under always is a bad person. That's not true. There are some fellows are also putting something in knowledge which is not applied in Google. Okay? Now, let me take you to the Western Ghats where I have been working for the last 200 years. None of you know. That is the environment we started working. You know, from Kerala to Gujarat, that is usually is 80 to 20 Now, you look at the Western Ghats. Because of 100 plus years, look at Kerala, there are 44 rivers in the Western Ghats. Because of this river, with the water, we have the food security rivers. Okay. Now let's look at our earth. You all know that we have more than one earth? I want to answer. We have only one earth. Now let us look at which part of the earth we have the highest number of plants and animal species. When we read the study, you see that this is the latitude or x-axis, y-axis number of species. You see, at 8 to 14 degree latitude, you have the highest number of plant and animal species. Highest number of plants and animal species means the varieties are high. Because of the varieties of things, you get different food. Because of the different food, you get the different medicine. That's why when people take the diverse food, they are very healthy. During COVID, I was working in Western Ghats villages. In Kerala, Karnataka, or Western Ghats. And we see people dying because of the COVID in those villages. It's only in Varthur, because here every other day somebody has died another day. That means the people in those villages, because of the availability of diverse food, they were having the diverse food every day and their body was immune to even the COVID or any zoonotic diseases. Okay? Now, if someone wants to study about plant, there are opportunities to study. We have 1,300 species of forest plant plus 250 species of birds which are issued which you and the secretary will talk about. They have updated the database. Okay, you can also take a president. Just try to understand any of the biology. What is the variety of birds and butterflies someday? And try to understand what they know, how they are useful to you. And doing this approach to see that each of the bird either giving you knowledge or helping you in some way of providing the food. When a bird and butterfly is inside, that's the most important job of pollination. When there is pollination, you have the food. If there is no pollination, you do not get the grain, you do not get the vegetation. So that is the importance of the pollination. You can also do the experiment in the region where pollinators are abundant. Go to the International Top side, look at what are the pollinators are there, and you go to another locality where there are no pollinators. Maybe they are there. Okay, you see that because of the pollination, how the food productivity has improved in the region, in the villages closer to the level. Okay, you can do that study. Okay, then try to understand the ecosystem function of each of the biological elements. Whether it's a cockroach or the mosquito or the thing. People say that they are harmful to us, but also try to see why they are there on the earth. If they are harmful, then why we have them on the earth? How they have evolved? What is the kind of thing happening in us? Okay, then you know the numbers. We are all part of this. We are thinking that we are exactly intelligent. That's our problem. When you have little intelligence, you will become more bad. If you have full knowledge, then you will not be the arrogant. How do you exhibit arrogance? Sitting in a classroom, talking around yourself is a show of arrogance. Or criticizing someone without understanding the subject is a sign of arrogance. Okay? So you would have seen at home, to just switch on the TV, the evening, news channel, you will see how arrogant people are doing this. Some illogical arguments will be happening. Okay? Now, coming back to the subject, you can also look at how the organisms have evolved and what kind of changes are happening. Okay? How they get adapted to the situation. If there is water crisis or the environmental issues. Okay? Let us also look at what is the importance of this. See, because most of you are from.
You got some project ideas from those slides? What did you understand? Who can summarize?
So there is a lot of diversity in Bangalore. There are 250 species of birds. There is rich diversity in Kerala.
That was Western Ghats what we spoke about. So Western Ghats is spread across how many states? Kerala, Karnataka. How many states? Give me a number.
There is a lot of species that help us to make things like medicines that
Basically different type of resources we harvest from these forests and keeping the habitat intact. Okay. And then in the last slide, what did he say? After Bangalore diversity. Mammals, evolution of different organisms, not just humans, different organisms. He showed humans because we are also humans. We are also mammals. Okay? And he mentioned one thing, right? In your area also you can study what are the different changes that are happening with the changing habitat. Okay, for example, your Varthur lake got rejuvenated recently, right?
Yes, sir.
So is there any changes that you see before and after?
Yes, sir.
What changes? Okay. I don't want to know. I will listen to you when you come back with your project. Okay, so you understood how projects can be done, right?
People will ask one question, what is the use of this? If you go to home and say that today I met a group of researchers from IISC. First question your parents will ask you, what is the use of that? That means every action of ours we will look at what is the use. Now let us do that. What is the use of this ecosystem? How do you do that? You find out what kind of goods and services are provided by the ecosystem in terms of food, energy, and medicinal value. Okay? Here there are some studies and now these are some of the studies. All know that because of photosynthesis we have oxygen. Now that is with the role of plants. What is the photosynthetic efficiency of plants? That can be an experiment. You find out how much carbon is stored in a plant, and how much oxygen is produced to get quantified. That could be done. Then storage and recycling of nutrients. The macro data essential for our body. You need the iron, you need magnesium, you need zinc. All those things they go to the doctor and they give this other. If you are naturally you will have some symptoms. Okay? Then the whole necessary habits of the detoxification, if we have so much waste, sewage, coffee, electric waste, how do you treat that? You can think about it. About the last days before here, 2 instances, we know water treatment technology. And also they said that how to monitor the water bodies using a robot. That is the beauty of your brain. When you are young, you will think positively. When you think positively, you come up with a very interesting outcome. See, most of the people who are successful today, they are achievements have happened when they are in the age group of, the 18 to 35 or 40 and 50. Okay? So, and then you can also look at what is the soil erosion. When we remove the vegetation from, how it affects the ecosystem. Then you can also look at the water resources, how much water is available in the region for drinking, etc. And how much is available you can compare with that, then you will understand how to manage the water. See, once you know that there is a scarcity of resources, your brain is not thinking of not wasting the water. Okay? Or any resource. Now, you can also see how people are connected with the nature. This gentleman's livelihood is met because of the extracting the sap from the various plants. That is the material of alcohol. These are the other benefits, You have the physical health. Because when you look at the plants, you will learn anything. You can be intellectually learning. Okay, emotional. When you are stressed, don't have a tree. It gives you the same level of comfort as when you come to your mind. It gives the same. You should do that, okay? Then, social health, environmental health. These are all the benefits you will get. Spiritual health. Why did these saints stay into the forest? They sat under the tree when they were talking about, thinking about the welfare of the society. Because they knew that if they are in that environment, they can come out with something interesting. When they interact directly with the nature. Okay? Then, what kind of food you should have? The food what you take also decides my country fate or my earth fate. If you start taking the food where the water footprint is less, then you have set the earth. For example, if you take pulses and millets, you are helping the system. It is a not resilient flow. The millet is not resilient. So, and also, when there are changes in climate, when there are droughts happen, millet changes as well. And millets and pulses require less water. And there are simple, what is the benefit of pulses? It helps in weight management. If you want to be slim and small, have no pulses nearby. It helps in brain function. If you want to be intelligent, make sure that your person will take pulses every day. Okay? It's a good source of vitamin and mineral. It also helps in muscle generation. If you want to look like Salman Khan, you can do that. Then the pyramid cell and gene damage. Just have a couple of pulses every day morning. Protect your pulses. Okay? It helps in digestion and boosts the metabolism. It reduces the risk of cancer. And also it lowers the blood pressure and it controls the diabetes. Now you might ask, what is the flow? See, one of my colleagues, look at his food plate, he has 5 items of vegetable, 5 items of food, and a whole person. Earlier he had the diabetes and blood pressure. Now he is about 75. Last 1 decade, he has practiced this diet. He either have blood pressure, nor the diabetes. See, the way you take the kind of food, that also decides your behavior. See, when you take the balanced diet like that, it rewards you physiological, psychological, sociological, environmental, nutritional, emotional, and spiritual well-being. The people who take balanced diet and healthy diet, they never get upset. People who are not only proper diet, you see them shouting at everybody, every meeting. Okay? Now let us, when you start having pulses and millets, what is the water footprint? Let us see. See, for example, to grow a kg of pulses, you need only 200 liters of water. To grow a kg of millet, you need 150 liters of water. To grow a kg of rice or a wheat, you need 3,000 to 5,000 liters of water. Now, to grow a kg of sugar, you need about 5,000 to 15,000 liters of water. Now, you see that when you start taking more pulses and millets like ragi and all those things, you see that you have reduced the footprint of the water environment. Don't you think that you are doing a great service? You should demand your parents to provide you the millets at home. And you should insist that they should, however busy they are, tell them that cooking food also for the children is equally important. That is their responsibility. They should do it. Okay? And more importantly, when you start eating pulses, we need to put pulses. When there is no pulses, there is no human animal conflict. In a region where rice is grown, the wild animal conflict is such the rice and sugar. But when you start growing pulses and millet, wildlife conflict can also be reduced. See, that could be another project. What is the water footprint of each other? Then, how to minimize the plant and the wildlife human conflict? That could be another. Okay. See, when you start practicing that, I will have healthy planet. When there is a healthy planet, there is a harmonious state of mind, body, and spirit. Okay, that is the holistic. Okay. Now the next environment you see is the pollution. Pollution.
Full summarize.
Nature is connected to the human, and without nature we can't live in the earth. And 71% is covered up to this water. And for Ragi we need to 3,000 water.
Basically, he explained about the water usage for different human uses, right from chocolate. So basically, the choices that we make in our everyday life, how much water, it's not just the water we take it as water, but also for the products that we consume on a daily basis, how much water is required. That determines the total water usage. That's what he tried to explain. After that,
Sir, lentils gives us many benefits. It gives us vitamins, minerals, and helps us to remove the cancer disease. And lentils help us by functioning the brain.
one more point he mentioned. The diet, yes, and also he spoke about avoiding one certain problem. Huh? Not health issues, ecosystem related issue. Very good, excellent. Human man-animal conflict also it helps reduce. Okay, can you proceed?
When you let the switch, what is generated in your toilet and the bathroom, if you do it, that is positive, you have to use it. The solid waste what is generated in your kitchen or in the home, if you dispose it out in the neighbor's compound or on the road, then you are polluting the surrounding. See, when you pollute your neighborhood, you have the problem of the vector-borne diseases. You can also study them. The Bangalore do not say to our school how to manage the solid, because every road has a solid. That's where I said the only waste people and waste practices are there. Okay? Then, yes, this is very important. Or when you dispose the waste into the surrounding environment, there are volatile compounds that get into it. You can look at this also. What is the air quality in Bangalore and major cities in Karnataka? There are monitoring stations, pollution control board has put the automatic sensors to monitor. You can compare the data, you can download the data and analyze to say which part of Bangalore is highly polluted. Okay? Then let us look at when you pollute the water body, what is the implication? See, every unit of pollutant getting into the water body, it gets accumulated in the fish to 1 million times. The fish, it's all eating many algae. Then every unit of algae gives common pollutant. Okay? That is where the problem of the pollution happens in the system. So now you can also study what are the fishes that are there in the ecosystem and what kind of pollutant it has, heavy metals and all those things. See, for all these experiments to happen, there should be a chemistry lab in your institution. Okay? Please make sure that there are chemistry labs in your institution so that you can do this chemical analysis. Okay? And molecularity is also our brain and should you all have that. Okay? So now you can see that. See, when you see a body in green, it shows there is a nuclear another movement happens. This is the clean water, whereas this is the dirty water. You can also do the water quality assessment. You can demonstrate how to do that. what are the variables to be looked at? She is going to talk about each of the variables and also she will shortlist the variables with the children's schedule of your age. Okay? So then you can also look at the interaction between the humans and environment. You know the forest resources, how the humans are dependent on the forest. If you are from rural area, you might be in agriculture, but you might be from the Uttar Kannada district or Shimoga district. You go to your grandparents' house, spend a week and try to understand what kind of forests are there, what are the species are there, how degradation happens, how to do weathering people. Okay? Then you might ask, by understanding this, what is the use? See, if you want water, you need to work for it. See, this is a photograph of 91 when I was a student and today. Look at the same region, I don't have water. That could be because of the surrounding area. Now I will the vegetation. You know, to understand that what we did an experiment. We put the sensors in an area. You know, the sensors will tell us how much water can percolate at every place. Okay? So we chose various landscape. You know, landscape covered with the native species forest, landscape covered with monoculture plantation, landscape covered with building, etc. And when we did the monitoring, we see that wherever native species forests are there, you know, the water percolation happens very effectively. That is mainly because when the native species forests are there, since the native species forests are the diverse, the roots are also there. When roots are diverse, soil microorganisms are there. When soil microorganisms are diverse, the soil microbial activity would be the efficient. When it is efficient, it makes the soil porous and permeable. When it makes the soil porous and permeable, it allows the water to percolate. That means when it rains, the water percolates, it fills this zone, which is a groundwater zone, then it fills this zone, which is a vital zone. Okay, then only you will see water flowing in the river or a stream or a lake. Now, you see that you have monsoon rain for 4 months. Because of that, you have water in the water body. Next 4 months, the water that is stored in the underground area here comes to the water body for the next 4 months. During summer, the water that is stored here comes to the river or stream or the lake for the next 4 months. That's how you have governments out there. Now, when you have buildings, you know, nothing goes underground. So there is no water. That's where the value of scarcity. We know that there are forests are there, water is everywhere. Our study shows about 60 to 65% of rainfall gets stored in an area. Whereas when the degradation happens, you see that there is only 20 to 25% of rainfall gets depleted. That's what the blue lines show. This shows there is more agriculture in the entire area. This shows where the forests are there. Okay, we also look at the demand of this water. You see, wherever the water is available, where ecosystem is there is surplus water is available, but the demand is much lower than the availability of water. Okay, to summarize what I said so far, you know, this is the region where government's water is available. This is the region, the 8 months, where this is 6 months, this is 4 months. Now you just look at the vegetation cover. You know, using remote sensing data, you have used the Google Earth. Now, next talk, the Google is going to tell you how to use the map, the spatial map, how to do this kind of analysis. She demonstrated that. When you overlay this on this, you see wherever forests are there, you have government's water. Wherever forests are fragmented, depending on the level of fragmentation, you have 80 seconds of water. Wherever forests are less than 30%, you have only 4 months of water. So now you know where is the water, how to get the water. See, now you might ask how this will happen. See, the farmers in this region, since the water is available by month, they can grow multiple crop for a cash crop. They earn 1,54,000 rupees per acre per year. Whereas farmers in this region, since the water is available only for 4 to 6 months, they can grow only one crop. They earn is 32,000 rupees per acre per year. This 1,54,000 is high and 32,000 is high. You agree with that number? Okay? Now look at the pollinators. See, you study the pollinators in this region, pollinators in this region. You will be surprised to see there are high density of pollinators here, diversity of pollinators. Whereas here there are few number of pollinators. Now you might ask, what is the rule? See, when pollinators are diverse and abundant, pollination becomes efficient. When pollination becomes efficient, yield of the crop is higher. For example, paddy, the farmers in this region, they get yield of the 14 to 18 quintals per acre. Whereas the farmers in this region, they get about 6 to 8 quintals per acre. Now you see how pollinators help you in your additional income. 6 to 8 quintals per acre versus the 14 to 18 quintals per acre. So that's where, see, when you do this kind of study, you will understand how to maintain the ecosystem so that everyone gets the benefit of the ecosystem. Okay? Now,
what's on this? You have done it. Somebody else.
Water recharge, pollinators growing the plants, water seeps into the ground, and plants grow well, and through that the pollinators pollinate plants and ecosystem will be good.
And what are those maps that you were using? You explain. Which area was that? This forest. Somewhere on the road. Sharavati Valley. Sharavati Valley. So what he showed there, one area has a lot of forest, one area has fragmented forest, and one area has degraded areas. So the difference in water availability and the pollinators in these areas, and how that led to the available water and also the yield of the crop. Okay.
we don't know the degradation is happening, what will happen to us? Okay, let us try to understand. See, these are the fish species we discovered in the western world. We can really do that, okay? But more importantly, they have medicinal properties. Then we thought, if we build to bamboo and cultivate it in our lab, we can multiply so that we can become big money people like Birla, Tata, Raman. But you know what this fish did? What we culture in the lab, they have the medicinal properties. So that means if you want medicine, you need to grow them in their habitat. Their habitat is this river basin. For a fish to survive in that river, it should have water throughout the year. So if there is no water, then that fish won't survive. Now for the fish to survive, to have 12 months water, it should have the forest in the catchment area of native species. This brings the linkage between the terrestrial biodiversity with the aquatic biodiversity. Aquatic means water, terrestrial means land. The forest, agriculture, all terrestrial system. Now let us also take another example, frogs. We call frogs as amphibians. Why do we call frogs as amphibians? One at a time. Now, one of you can try it. Is the man is there? When you learn, you should understand and learn. You should never just jump to the conclusion and make a statement. Those people who just get through the exam by mugging, they will never succeed in anything. But when you read the subject carefully, you will understand the concept. Now, I will answer the same question in a different way. Frogs have two stages in the life cycle. Juvenile stage or a tadpole stage, which is in the water. The next stage is the adult stage, which is on the land. Because of the two stages in the life cycle, requiring two different kind of habitats, they are known as amphibians. Okay? Now, if there is no water, then what will happen to frogs? Tadpole cannot survive. In such a situation, when there was no water, one particular species of frog which we come across in our study is divided as mucorales. We saw that after fertilization of the egg, on the 14th day we saw that intervals energy out. Because we are going to the forest every day. On the 14th day we saw that was coming out. We were surprised. How did this happen? Then we got those fertilized eggs to the lab. And when we frogs have learned how to get adapted to a situation where there is a water space. Now, as you all know, we have only one earth, which you also told me. That one earth, if we lose water, no one will survive on that earth. Only these organisms who have got that adaptation. There is this is another experiment we can think about it. That's where I said, when you start observing nature, you will be surprised. I will give a simple example. If you have another at home who is writing, just look at their work, what they do in the day, assign the value to it, and find out how much, what is the work of mother or dad and working in the house. Okay? So, what is the now, see, understanding these kind of behaviors, our elders protected the culture. They are not sacred to regular Prabhu, Tavu, Tan, depending on life. You can go on, Tavu, Tan, okay, like that. Okay? Unfortunately, what we are doing, we are converting the forest into idle. Idle is always thinking. Okay? That is, we are doing the okay? Now, let us also look at what are we doing the way forward. Look at what we are doing. One vision, 2,700 liters of water is required. Okay? You only eat chocolate, I don't say you should not eat. Per kg of chocolate is around 2,700 liters. Okay? Then look at the coffee. 1 cup of coffee is about 1,000 liters. You take it from the way of boiling coffee and all those things. Okay? And look at the favorite food. It has the food, the water footprint of 2,400 liters. And from 180 liters. Okay? Now, you can also do the kind of water footprint. The second thing is the cultural footprint. We all take the food and everything we do. We start and then do vehicle, etc. Come to the cultural footprint. You will be surprised to see. What is your footprint? For example, you see United Nations for every individual 9.6 hectare of the land resources they take for. Whereas in India it's about 0.8 hectare. Don't tell me, no, we should become like America, we should also have that. That is the responsibility behavior. Becoming more and more consumer is not a good sign. Because that's where optimization, prudent management of resources are required. Okay? So now let us take up some example. How do you map the education? I have two youngsters from Jain University here. Ganesh. Gagan and Ganesh. Okay? So, we have who is going to teach you about the vegetation, and we have Bhagana who will teach about water. General education. Okay. Now, the business. So, they will help you in identifying the plants. They take those five. Okay? In case you have a window, you can consult. They will tell you how to identify that. And even we are not going to do that. Today we have become a busy. As we age, we start helping them. Now, when you do this kind of vegetation management, say for example, when you find out this is in one village, which was grown in England. This is enough to allow you to get a variety of rice. Look at that. Each one is known for fragrance, one is for eating, one you can make good dosa, one you can make good the rice, or in biryani like that. Okay? And these are the other words. See, that is what you should study. What is the kind of varieties of plants you have on in your system? Okay? So, and you can also do that.
Who will start with the fish? Every time you both are doing it. Show us a bit of it. Where the water is there, we get fish.
Water is sufficient for fish. This is very important for species.
Okay. He told something about medicinal properties in fish. When the fish was taken from the stream, it had medicinal properties. So when we brought to the lab and cultured it, we did not have the medicinal properties. So what was understood? It will have medicinal properties only in that particular habitat. Okay, then. Okay. Okay. No, tell me about the first thing. Now they have started adapting, skipping the tadpole stage basically. Next. Sit. Continue. But here, this case study, what was observed is that species of frog is skipping the tadpole stage. From the egg, directly the young ones of the frog are emerging. The small froglets are emerging. Then after that, somebody from the last. Who will explain? You're looking at the book. You can see the book and answer, no problem. Very good. Okay. Frogs and what was the last slide? The slide before this. This row, except you, nobody will answer it. Can you help me? No. This one. So what did he say in that slide? In your region, you can identify the different types of, not just rice, any food items. Okay, good.
So naturally, that is where you need this GPS, O, pay, etc. and the situation, etc. to do that. Okay. Then you can select your village or the what. You can get this map from the village accountant office. Today this map, the account map is available online. Okay. So, and also in the book, you can view your village, you can view your school, you can view your the what. Okay, you can also do something online. Our tutor is going to explain how to do that. Okay. So, and when you select the village, see, when you do the mapping with the help of pencil like Gagan and Ganesh, you can also develop the mapping. This each silver, color silver, is the plan. And since they can identify, they will also tell what is the species or type of plant is that. Okay? So then, this is the close-up of that. Okay? Then when you do it, you can assign the color to the various the type of plant, and you have the varieties. Okay? So now this is another way. Okay? See, let's look at plant. We want to find out what particular carbon that is stored in the plant, you need to know the biomass level. Then how do you measure biomass? There is completely varied. But that is a distraction. You should not cut it. Then what is the other way? You measure the depth of a tree and height of a tree. Once you have the depth and height of a tree, you can find out what is the volume of the tree. Simple cylinder example. You know the cylinder volume? What is the volume of the cylinder? Yes. Very good. The volume of the cylinder? Very good. The same thing you will use it. Okay? Now, how do you measure the height? See, you have eyes, very beautiful eyes. You make use of it. Okay? And use a compass. Suppose if you don't have a compass, use your paper, 45 degree, 90 degree. At a horizontal distance, you look at the tip of the tree. Then you start moving the backward. So that means you have known horizontal distance of the tree and you know the tan and the tan theta. So once you know the adjacent side and the tan theta, you can find out the opposite vertical. Okay, this is the symbol tan theta. Okay, you can do that, you will get the height. So once you have height and depth, that is the circumference, you can find out the radius of the tree and you can find out what is the volume of that. With the density of that load, you can find out what is the biomass. And about 47% of the biomass is the carbon. Okay? So now you can also do the sampling like this, 1 hectare plot you can take. If the region is small, you can do the censusing of the entire region. Censusing means each tree you will have. Okay? Then, if the forest area is big, then you can have a transect-based quadrant. Okay? You will have a quadrant. Within the quadrant, you will find out the trees, the 20 meter by 20 meter. Within that new quadrant of 5 meter is there, which starts on map. Within that, there are 1 hectare quadrant where you have grasses and crops. Okay? That's how we map the trees, shrubs and stuff. Those who are interested in trees and herbs and shrubs, I suggest you to talk to me, Gagan and Danai. We will explain to you further. Okay, then once you have done the record, locality, the platform of that, and also how to use GPS, we will explain to you, and how to take photographs using that your mobile, you can have the GPS camera. Okay. See, this is what children have done. This is almost the 15 years back. Ali, she brought the student to our campus. The student did the measurement. Each child measured the depth of every and height of it. When a class of 60 to 70 kids do this experiment, you will get 60 values for the same tree. Because each child does the measurement differently. One fellow gives in meter, one fellow is centimeter, and that kind of variation. See, you record it, the student one. You can also teach statistics, find out the mean and standard deviation. When you are teaching science, you are also teaching the mathematics. This is the best way of teaching the subject so that children learn more effectively. Okay? When they do this kind of thing, they also have variation. But it is allowed them to control the system by doing them. But they are enjoyed. They have a good food and they went around, they enjoy the campus like that. See, this is the only subject. It gives you the physical recreation as well as the knowledge. Okay? Then, wherever lakes is there, you should rub the lake, you monitor the lake, catchment mapping, then you can collect the water sample, you can do that. See, the children are trained by my student, the Varthur, you can see how they are doing. And then children go and collect the sample, then they go to the lab, they analyze it, they map these algae, diatoms, etc. These are all done by the fish. You can ask the fish who did these sketches, where are they? You will see their document. Okay? So, and the same, there is a diagram when you see to the electron microscope. You see. Look at this structure. You will get a lot of ideas. This organism, every 4 hours fire goes on reproducing. Within 4 hours fire, the synergy. And whereas we have to design a product like this, it will take long time. You know, we have tea break, coffee break, and all those things. But nature does it without any break. Okay? So, and this diagram also has oil. You can have the biofuel also. Okay? And the children, when they do the mapping of biodiversity, you know, they come, they map, we validate for the student. This is what we do with the children in Shimoga, Uttarakhand, etc. Okay? And this is one of the other areas where you can see the thing. You can see a municipal is there, and the politicians are there, local sales are there, and experts are there. Okay? That is what the problem is. Okay? And if you need more details, okay, you can actually at this email. And this is our webpage where all the details are available.
Please
We do not use mobile because I should be happy because we use mobile. Okay? And also I don't have time to waste for me. I feel every week matters to me. If I can spend one day with one student, I've done something in my life. Okay? That's why I avoid using mobile and all this artificial. I believe in nature, I believe in nature with real energy that we don't know. Okay? Right. When you look at the website, you will see all my publications and also available. Okay? Then, you can also take up the physical experiment. What is energy consumption across the various varieties of clouds? Any what sort of interaction was with the person, two lights, then you compare conversation. You can what is the light and hours. See, you can try to see the kind of brightness what you get. The same brightness you can get from the rain. Instead of using 45 fluorescent lamp, you can use 10 watts, 15 watts creator. And look at the number of the time there is a lifetime. You know, for every 15,000 watts, okay? Whereas the incandescent 1,200. That means in a lifetime of 1 LED, you have to replace incandescent bulb 50 times. Okay? And 8,000 is a compact bulb. Now, look at the energy requirement. For LED, it's about 8 to 10 watts. The incandescent bulb 60 watts. And see a person, you see the amount of energy you are going to transfer. You get the same illumination. Okay? And light is also longer. See, that is what sustainable. When you use it, you will also have lesser waste product in the system. You will have 150 bulb and throw it in the environment. In the one LED bulb. See, you can do this kind of experiment. Water harvesting, energy harvesting. And where minimize the wastage, you can think about it, okay? Then you can also look at your use paper, okay? To manufacture paper, what is the energy footprint and the water footprint? Don't just search and do that. You also carry out experiment. Try to manufacture paper. Look at what is the footprint, okay? So now, you know, the people have their own, one metric ton paper becomes 70 Gs. Okay? The 24 Gs are white office paper. You can see every time when you waste paper, you can see what is the key footprint also. Okay? And how much electricity? 10,061 units of electricity you require. 10,000 units. Okay? You require 2.57 cubic feet of oil. You all have water tank at 1,000 cubic feet, 1,000 liter, 1 cubic feet. 2.5 times water tank, the oil will be consumed. See, that is where when you learn this thing, you will never think of wasting chemistry. Because you know that wastage will make you, that tomorrow you will not have the resources. Okay? So, now that is what the knowledge we bring along with children. When you put this knowledge in your any way, if you take it slowly and learn the concept, then you will be a highly successful engineer. We are looking forward for that, okay? And look at this kind of thing. There is a water body in our campus. This is the lab for our students. I have told in BTS also there is a water body. But we have chemistry lab for the children. So BTS should never worry about the they did this mapping, how the lake was there earlier, and everywhere how it is, then what is all lakes with planters, fish, etc. They mapped it. You can also look at the similar thing for the lake in these planters, okay? And you should tell your principal. Lake is for 1,000 children. When you have 1,000 children in this campus, the lake is also equal income. You should also manage that, okay? So, see, when I meet people, I also make them to create a water. This is what we go with the same memory. The next month, he has to create a water body in his campus. Similarly, I had another young man here. He is the head of our service. Okay? In his campus, he created a water body which was present at years. And he also take up the tracking of native species in that campus. When we visit the campus, myself, Vishnu, and now quite often, we see greenery with the water body here, both resources. That boat is because of the when many people are there, they do this kind of religious things. When people get old, they become, sick. So let us not worry about old people. They are out there. Let us think of any people or any people who is dead or something. Okay? See, you might say, what is the use of this? What is the other? This water body was created in 2016. He did the research of 2 days. Because he did the research of 2 days, 30 days in that particular was the research by the community. When all others now face water crisis, this particular water does not face water crisis. That means this intervention has helped in the providing of water. That's what the religious leaders should do. They should ensure the sustenance of natural resources like water, the vegetation, etc. When they do that, there will be prosperity in our country. That's where I think telling and I write. Science, society, culture and spirituality should go hand in hand in our country to see the prosperity in our country through the sustenance of natural resources. Okay? So then, if you come to my campus, you will see this campus. When I was came, I was fascinated by this campus. I was directly to the campus. I was a student at that time. I brought the seed to the campus, planted in 7 locations. Out of 14 7 seeds were planted in 7 locations. Out of 7, only one has survived, that is next to the village where I got for 2 decades. That means even the plants know who will take care. Because other 6 places, which is far away from this village, they didn't survive. That means even the plants know who will take care. What do you say that you are doing? Right? So then, in front of the city, we created a mini house. 49 native species, 5 samples were planted in 2 hectares area. At that time, groundwater area was 150 feet. Today, I have over 10 feet. Not only that, I have a lot of friends, 4 families of children, varieties of birds and snakes. We are not present in the snake. When we see a snake walking there, we go on up to them. There are instances every day where I will after lunch when I walk in our evening one level comes there. I always think that he has come to see me and I have come here to see him. So that's how we reciprocate the relationship and respect to one each other. Okay? So now the temperature in that building for us is 2 degrees. That means the vegetation and water body help in moderating the microenvironment. Okay? So now that's how we communicate to the wider section of society. Now today we are doing the same thing here. And also for the people who are seniors, who are working in industries and the universities, etc. We operate most of the economical management. Now I was told Galan and Ganesh also want to do the course. That's how they have come here to talk to me. Okay? So then, you are all welcome to Lake Symposium. When you do a good job, like the lecturers who work with me, this is my availability. Thank you very much. I'm sure you take it the same forward and see something great happens in the Lake Symposium. You are all invited to Lake Symposium. You come there to the building, you will take care of your accommodation and food. You will enjoy. And you will see how we also work. That might give you some ideas to you. Even if you are not selected, you are welcome to come here. Okay? And India said, when we talk about the good for us, we want more and more excel in company and success. If all of you do a good job, I will select all of you. So I want each one of you to pose a challenge to the people who evaluate your performance. And when you start doing it, you will see the real development in our country. You know, when children start acquiring additional knowledge, we have the
Nature takes its own course of action, because usually when we start research work in the field, we start with the birds because they are very early than other organisms. So many of us are lazy to do bird watching because we have to get up very early. With the first light, the birds start becoming very active. We hear the birds in the morning, right? So I have been researching birds for more than 10 years now. And whenever we have to do field work, we have to wake up very early before that and be in the field by that time so that we can start observing them. Okay? So today also they have arrived first and we'll start with the birds. So how many of you have done bird watching before? Nobody. Excellent. Have you seen some birds around you? Yes. Crow, Myna, pigeon. Thank you. Eagle, where have you seen? I'll come back to you. Okay? Okay, so can you tell me what are birds? But what are they? Can you look at me and call me a bird? Then flying creatures and that have wings. Mosquito is a flying creature with wings. Very good, they lay hard shell eggs. Very there on people. That's it. They have feathers, very good. That is something that is very unique only to the birds in the entire animal kingdom. Okay, no other organism have feathers other than birds. Very good. Then beak. Beak is also something that is very unique only to the birds. No other organism in the animal kingdom has beak. Okay. Claws. Yeah, they have claws, but not all birds have a prominent claws which can hold on to other organisms. Okay. So let us look at it. Now can you describe a bird? You mentioned wings and feathers and hard-shelled eggs and beaks. They are also warm-blooded just like us. What do you mean by warm-blood? Bit hot. And what is cold blood? Which is cold? Yeah. So butterfly has, or all insects and reptiles have cold blood. Even fishes, which since we will talk about, they also have cold blood. So what is cold blood? Which means that their blood is colder. Right? See, one simple example I will give you. What is your body temperature? 98.6 degrees Fahrenheit. Okay, and you said that's a big range. 36 to 38 degrees Celsius is the normal human temperature. If it goes higher, then what do we say? We have fever because it is not a normal temperature. That is mainly because we have a constant body temperature. Okay? So any organism that has a constant body temperature will be termed as warm-blooded creatures. Understood? So birds have a constant body temperature. Now don't ask me how much because it varies from species to species. Okay? So what is cold blood now? Any organism that does not have a constant body temperature can be termed as cold-blooded organism. So what is their body temperature then? It depends on their environment. Okay, for example, a fish that lives in water, whatever is the temperature of the water will be the body temperature of the fish. Same with the amphibians also. Amphibians, reptiles, all of them, it goes the same. Any cold-blooded organism. So cold-blooded organism usually need sunlight to warm their body for starting their bodily activities. Is that understood? And the advantage of having a cold blood for an organism like a crocodile is they can lower their body temperature and lower their metabolism rate because of which with minimal amount of food they can extend their life for a longer period. Okay, in Africa where there are drought conditions, a crocodile, if it has its stomach full of food, it can go up to a year without eating again. Okay, it can slow its heartbeat like 4 beats per minute only. It can reduce it that much and live for a very long time without food. Can we live for a year?
No, sir.
Definitely not even for 4 days. Right? So now, one of you said birds evolved from dinosaurs. Whenever we think of dinosaurs, because of the movies like Jurassic Park and Jurassic World and all that, we imagine them to be really big. Okay, man-eating and they come chasing and all that. But that is not true. Not true as in they are carnivore, and if it comes, that happens. But what evolved into birds are not the large ones. The tiny, small dinosaurs that existed, they were able to evolve into birds or other organisms. Okay? So, see here what happened. The dinosaurs, which did not have wings, had a certain geographical area. For example, if I ask you all to walk how much ever you want, how much will you walk? A few kilometers only, not more than that. Right? If I give you a bicycle and ask you to travel, you will go slightly further. And if I give you a motorbike, much higher, you might even go out of Bangalore. Right? If I give you a car and say go wherever you want with full tank fuel, you will roam further. And if I give you a helicopter, you will travel around the globe. Right? Same thing happened here. The ones that did not have wings, once they developed wings, they became highly locomotive. The ones that were restricted to a small habitat started crossing oceans and mountains and going to new places which they have never ventured into before. When they went to all these new places, they were exposed to new habitats and new food items. Right? Is the world same everywhere?
No, sir.
It's definitely different, right?
Yes, sir.
So when we go to different places, we have to adapt. For example, if you go to Himalayas, can we wear clothing like this?
No, sir.
If you go to Thar Desert, can we wear clothing like this? No, it has to be according to the geographical conditions and the climatic conditions there. So these organisms also, when they venture into a new habitat, they started getting accustomed to those kind of conditions. So eventually what happened, one species when they spread to different areas, they started making different adaptations. And with those adaptations, they started becoming different species altogether. Understood? That is how speciation happened. Even now we see this trend happening. For example, you have seen commonly the crows here, right? The fully black crows. They were called the Indian jungle crow and they had the scientific name Corvus macrorhynchos. Macrorhynchos means big beak. Compared to other crows, they have a larger beak. Okay? But now, from 3 years, they are separated as a species where what we find in the Himalayan ranges are called the Corvus macrorhynchos and what we find here are Corvus culminates. Okay? Basically, they found genetic difference between the species. It is the same species before. First, they got divided as subspecies. Then, over a period of time, there is so much of difference between the two that they can be considered two different species. Understood? So evolution is a continuous process and it keeps on happening as long as there is life existing on the planet. Okay? So next. So like I said, they went to all the different places across the globe. Wherever you go, you will definitely see birds. Conditions apply is depending on the season. Okay, next. So with the speciation, don't write all of this down, just write this last part. Today, we can see more than 10,000 species across the globe. Of which order? Which organism? Yes, birds. So close to 11,000 species of birds have been documented across the globe. Actually, it is 13,000. Many are extinct. Okay? So alive are these many numbers. Understood? So how many are there in India? Yes. Huh? No, it cannot be uncounted. We have counted for the entire globe now. So for India, it is definitely counted. 2,000 something, okay? 4,000. 1,500. You were there on that session. That's why you know. Yeah, it is around 1,500 species have been documented in India. In Karnataka, you have brought the same book, is it? Different book. Okay. About 528 species have been documented in Karnataka. In Bangalore, 200. 253 species in Bangalore, and in Varthur, 3. In your campus, I can find more than 3. I don't know. You have to tell me. So you can work on this project, and when we come in July, you can present about this and showcase to us about how many species of birds you have found in Varthur area. Okay? Alright. So this is an organization called IUCN. IUCN means International Union for Conservation of Nature. Okay? International Union for Conservation of Nature. This organization, every few years, they conduct a survey and then they assess the population trend of a particular species and then they give different status. So not all statuses are mentioned here. It starts with least concern, data deficient, least concern, then vulnerable, threatened, no, near threatened, threatened, endangered, critically endangered, extinct in wild, and extinct. All these are different status that are assigned by the IUCN. So we will not worry about the extinct ones because we cannot revive them. But what we will worry about are these, where critically endangered, today we have about 221 different species across the globe. So if any critically endangered species or endangered or any of these from near threatened onwards, upward, any of these, then wherever you find that, that particular space becomes an important area for conservation. Okay? Least concern means it does not have any threat currently, the population is stable. If it is near threatened, it is getting endangered. Right? So some action is required, some intervention is required to conserve them. So if you find anything above this, it means that that area is important for conservation and measures have to be taken for saving that particular species and its habitat. Now when we say conservation of species, don't think that we have to catch that, bring it and put it in a cage and protect it. That is not conservation. That we do for certain species which cannot breed successfully in the wild. Okay, so that is when it gets the status extinct in wild, but in captivity it is still there and there is a possibility of reviving that population. But here what we are doing here is we conserve the entire habitat so that the bird or any other organism which is in these status can survive on its own and revive its population. Understood? Okay. Next. So why should we study birds? So what? You know their habitat, their adaptations, and then what? They are beautiful, so what? Very good. Useful for farmers because they do biological control. For example, how many of you have seen owls near your homes? Owls. And have you heard your parents or neighbors or somebody say it is apashakuna? Yes. It is bad omen if they come and sit on your building. But please go back and tell them that is wrong. If they are there, it means that there are a lot of rodents in your area, which is rats and bandicoots, which I am sure you have seen that also. They will help you in controlling the population of rats and bandicoots and preserve your environment for you. Okay? So actually they are bad or good? Good. They have some ecological importance. So they have a series of ecological importance. One is biological control, which we just discussed. Then they are present at different trophic webs. You know, you have studied food web and food chain, right?
Yes, sir.
Interlocking of several food chains are food web. So birds are placed at different levels of the food web and trophic levels, where birds feed on different varieties of organisms. For example, some birds like sunbirds that you see in your campus feed on nectar. Okay, directly plant product. Then there are some birds like barbets which feed on fruits. Okay, fruits and berries. And then like parakeets that eat the fruits that we eat also, like mangoes and jackfruit and things like that. Then there are other birds that eat the insects. Then there are birds like vultures that eat dead animals also. So they have different roles in the ecosystem, right? So if a particular species is removed from the ecosystem, there will be an imbalance there and the entire ecosystem may collapse. So we can look at them as bioindicators. Like how your vehicles have indicators, right? If the right side light is blinking, what does it mean?
Indicator.
What does it indicate? Yeah, it indicates that the person is going to turn right. So like that, these are bioindicators of a ecosystem. Okay, looking at birds, we can understand the health of an ecosystem. They indicate the health. For example, if you see an area with lots and lots of pigeons, only pigeons, what does that indicate? Is it a good ecosystem? No, because there is a domination by one particular species, whereas an area with a mixture of different type of species is an indication of good ecosystem. Understood? Okay. Okay. This part, there he said, right, it helps the farmers. It also does pollination and seed dispersal. See, I said some birds consume nectar. So while consuming nectar, they pollinate the flowers, which help them produce the fruits which might have the seeds inside. Okay? Now I'll give you one example here, which is a state tree of Karnataka. State tree. Is there a state tree also? Is that what your expression means? Yeah, there is. Every state has a state tree. And Karnataka has a state bird also, and butterfly also. So which is the state tree of Karnataka? Yeah. Very good. It is Indian sandalwood, Santana Marble. Shrigandha. In Kannada we call it Shrigandha. So that is the state tree of Karnataka. Now if we remove birds from the ecosystem, this tree also will disappear. Okay? This is mainly because sandalwood produces fruits. Fruits will have seeds inside. Okay, so if you take those seeds and put it in a pot and wait all your life, it will never germinate. Because the seed has a coating which needs to be chemically processed, and only then the seed can germinate. You know where is that lab which processes it chemically? It is in the bird's stomach. So when the bird eats the fruits of sandalwood, the fruit pulp will be digested, the seeds will be, the coating will be processed with the digestive juices of the bird. So that is the lab. And then later when they excrete it, the seeds will come out as it is, and that is when they are capable of germination. And it's also a root parasite, which means that they cannot absorb nutrients from the soil. They will absorb the nutrients from the roots of other trees or grass or plants or anything. Understood? So without birds, several species of trees like this will become extinct. So birds play a major role in the ecosystem, in maintaining the health of the ecosystem. Now I explained about the ecological significance. Now from birds, we don't have only ecological significance, we also have technological significance. This is one such example. There are several examples, but we don't have the time for discussing all of them. There are YouTube videos in our channel, which I have given a 1 hour session, where you can see all those examples also. So here, this is a quick one. Bullet train, you all know? Yes, this was invented in Japan. Okay, so when they invented it, it was built for speed, right?
Yes,
And it was named bullet, so it was made, the engine was designed in the shape of the bullet. But this had a major problem. It had to pass in that high speed through several tunnels. And every time it passed through the tunnels, it created a sonic boom, like how bullet comes out of the barrel of the gun. Imagine the train the size of the bullet and tunnel being the size like the barrel. So the sound can be very explosive, right? But here it was more of a sonic boom. Okay? So they were breaking their head on how to solve this problem. And luckily one of the lead engineers happened to be a bird watcher. He was not a professional bird watcher, he was a hobbyist. That's all. So on his off day, he was sitting by a lake and observing the birds. And initially he saw a kingfisher come and dive into the water, catch a fish and fly away. Most of you would have seen that also. So what's so special about it? He thought life of a kingfisher is so much easier, right? It just has to eat and sleep and its job is done. But when he stayed there for some more time, he saw kingfisher taking the fish again and again, and he noticed something very interesting. Every time it dived into the water, the beak enters first, right?
Yes, sir.
When the beak enters, there is no splash in the water. Then which organ enters? The head. When head enters also, there is no splash. Only when the wings hit the water, there is a splash. So he got an idea, he called his team and redesigned the entire shape of the bullet train in the shape of the Kingfisher's head. And then when they did the test run, there was no sonic boom at all. So Kingfisher has evolved this strategy with millions of years of evolution to catch the fish. Fishes, how many of you have aquariums at home? Okay. If you have aquarium at home, you can observe the fishes have a lateral line, a dotted line from the gills all the way to the tail. Okay? That is a sensory organ which helps detect any movement in the water. Understood? So for example, you throw a stone into the water body. The moment stone reaches the water and creates a wave, the fish need not think, okay, I have to move, there is a stone coming. It will automatically move. Like how we all are breathing. Are we thinking, okay, I have to inhale, exhale? It is happening automatically, right? So same way, the fishes don't have to think before they move from a disturbance in the water. Understood? So with this adaptation in fish, it becomes very difficult for the bird to catch the fish. That is why over millions of years of evolution, Kingfisher has developed this strategy where without disturbing the water, they can enter the water body and catch the fish. Okay? Now how we have made use of it? To the bullet train, but we never acknowledge and say thank you to Kingfisher. Next. So now you are all interested in birds, right? When you start observing, for 1 day you will observe, 2 days you will observe, 3rd day you will sleep. Why? Because you see so many birds and you will not know names of it. Starting 2 days you will scratch your head and try to search and all that, then you will say I will not find it, so give up on bird watching. That usually happens. So I will teach you how to identify birds. You have to become familiar with certain things which helps you narrow down your search. There are close to 11,000 species in the world, right? And if you have to identify one species, there is a lot of narrowing down to be done. Okay? So how to narrow it down? We will see. And remember not to search for penguin in Bangalore. Because there are a lot of people who go into the wild saying that I want to spot this particular bird. They don't want to see all birds that are there in front of them. They want to go in search of one particular species. Right? Not just penguin, but any. So when you go like that, you should know where they live. Without that, you can be roaming all other places without finding it at all. Okay? Next, one important tip for you, don't worry about all those things that I have written there. Okay, that's not important. What you need to remember is, birds have been given names based on their appearance. Okay? For example, the bulbul that you see, they have red color here in the cheeks, right? So that is the whisker area. So it is called red whiskered bulbul. Then there is another one called red vented bulbul, where under the tail they have red color. That is the ventral area, so it is called red vented bulbul. Like that, everyone have been given names based on their appearance. Okay, most of them. So whenever you find a name of the bird, try to relate to its appearance so that you will easily remember it. Next time you see the bird, you don't have to search. Understood? Otherwise, what happens? If you come with me for bird walking, like if you go for a walk, I'll give you 15 to 20 names. By the time you go home, you will mix the names. So you will create new hybrids. So we don't want that. So first what you need to observe is the bird's size. Okay? So for example, he will go for bird watching near his home. And then he will say, sir, I saw a small bird. Okay? How many small birds are there in the world? And how do I know how small he has seen? So the small he is describing and the small that I am imagining might be different, right? So it will be difficult for me to help him identify. So what we do, we compare it with the common birds that we know. These 4 birds mentioned here, you all know these birds, right? Anyone who doesn't know sparrow, myna, crow, eagle, all of you know. So when he comes and says to me that, sir, I saw a bird of the size of the sparrow, we both will be imagining the same size. Right? So that way, it will help us narrow down accurately to the species, probable species. Next is, you need to mention the color. Which color the bird was. For example, if you have seen a crow. Yeah. Similarly, if you have seen a kingfisher. See, different people giving different colors. Why? Because you have seen kingfishers from different angles. Yeah, there are a lot of birds here, but everyone cannot see. Later you can see them. So kingfisher, if you see from the back, the major color that you will see is blue. If you see it from the front, the major color you will see is orange. Okay? So that's very important, from which angle you are seeing the bird and what is the major color and minor color. Okay, sometimes these birds have small markings, dots and all that. So that if you are able to see, please note down that also. That will help us narrow down the search. Okay? Next, these are bonus points. Okay? The previous slide, whatever I explained, those are mandatory points. That is the shape, major color, minor color. These three are mandatory to narrow down the search. Then whatever I am explaining now is a bonus. If you get to see the bird a little closer for a longer time, you will be able to see what type of beak it has. Looking at the beak, we will be able to understand what they eat. Okay? See, the first one is a generalist beak which crows and mynas and pigeons have. All the others are specialized beaks. Okay? So they perform a particular, or they consume a particular type of food, which is why they have a certain adaptation in their beaks. So when you look at the birds and if you get to see them up close, please identify the beak shape also. If you describe that, it will help us narrow down the search furthermore and easily identify the species. Okay? And if they come really close to you, only then you will be able to see the feet adaptations, the different types of feet. By looking at their feet, we understand which part of the habitat they live in. For example, there are swifts and swallows, which are not here. They have very tiny legs. They hardly spend time on the ground. They spend most of their life flying. So that is why they have very tiny legs. But if you see herons and egrets and all those, they have a longer digits. These finger-like projections, and they are extremely long because they walk in between the vegetation and wet sand. So they should not sink down, right? That is why they have that adaptation. And if you look at a sparrow, can you see the nails? We call them talons. They are very long. Why? Because they have to even hold on a grass and stand. You have seen sparrows standing on grass, right? Yeah, so if they don't have this adaptation, they will not be able to grip on those delicate habitats. Next, see the woodpecker and parakeet. They have two forward, two backward. This is what enables them to hold their foot like this or grip on a vertical area. Understood? Then duck I need not explain. You all know from nursery you would have studied. They have webbed feet. Then eagle, can you see the digits are bulkier compared to others? Because they have to carry weight. They carry other animals, right, for their food. So that's why they have bulkier digits so that they can easily carry. Then not always you will see a bird sitting and posing for you, right? Do you see them? If you say, I want to observe you, come closer, they'll come. Sometimes you might see them flying also. So then you have to observe their wing shapes, and with that we will be able to narrow down our search. Understood? Yes. Then some birds, male and female, look same. For example, here the Ashiprinia, the male and the female are identical. You will be able to tell the difference between the two genders only if you spend some time observing the two and if they are doing courtship. Courtship is just before breeding, they do some dance and singing and all that. So if you see that, you will understand, okay, the one that is performing is the male, the other one sitting and observing is the female. But otherwise, they look the same and it is difficult to tell them. Even for me, it is difficult. But in some species, the male and female are totally different. For example, Kogile, you all know. Quail. So the male looks completely black with blood red eyes. The female looks brown and white with the same type of eyes. If you see them in the field, you might even mistake it for a different species. But they are the same species, but the gender is different. Gender is different. Okay, so this you need to keep in mind because when you start observing the birds, this happens. So generally you will say, sir, I saw a bird which I don't know what it is, I could not find it in any book. Because most of the books will give you the pictures of males. Okay, females of several species look the same. So that's why you have that identification problems. Then in some birds, see this is a bird called rosy starling, which comes all the way from Turkmenistan to India. Every winter they come here, okay, without any passport and visa. When they come here, they'll all be like this, like bald. Okay, once they come here, within few weeks, the males that are ready to breed will get feathers like this. They are trying to impress the female. Okay, so that's why they get those extra crests. So at this point of time, if you see them, you will be able to easily differentiate between the males and the females. Otherwise, they'll all be like this. So it is difficult to tell them apart. Okay, next. Then this is a resident bird. Throughout the year, you can find them here. Throughout the year, he will be like this. During breeding season, which starts from January, he will turn into like this. He will develop golden brown feathers and look very beautiful to impress the female. Right after the breeding season, all those feathers will fall off, and then he will go back to looking like this. Why this adaptation happens is, see, this bird is a ground bird. Nearby to water bodies and grass, they will be spending most of their time, and they feed on something that is below them. Insects below them. Okay? So when the insects are looking up, what is the major thing that they see? Very good. Who said clouds? Very good. It is the clouds or the sky. Okay? So by being white, they merge easily with the cloud or the sky. So it is difficult for the insects to spot them. But if they are like this throughout the year, they cannot get food efficiently and they cannot become strong. Okay? That is why, but they have to impress the female also. So if all males are like this, can they impress the female? It becomes difficult for the female. So what they do, only the strongest ones will develop more beautiful feathers. Okay? Which means that is an indication to the female saying that this male is very strong, he is capable of finding food and providing for the family. Understood? See how much they also think. Next. Then the other thing is, there are some very interesting behaviors in birds. Okay? When you go for your project work, you start observing the birds, right? Don't go here and see a crow, right crow, look for the other bird. Spend some time looking at the bird and identify what it is doing. And if you see some interesting activity, please write that down also. By doing this observation, we also notice that some crows in some areas in Bangalore, they are stealing hangers and clips from the terrace. You might wonder some neighbor is stealing it, but it is actually our birds that are stealing it, because they have understood these plastic materials provide better insulation for their eggs. So they have been stealing. This is an interesting record that is happening, and we are observing that. Okay, so similarly, you might also find some very interesting behaviors which you can spot only when you observe. You have to spend some time observing their behavior. Okay, so you will see them feeding, nesting, then all these are feeding. Then courtship, different birds have different courtship. Some sing, some dance, some sing and dance. Some do acrobatics in the air. Then some have to do some stunts. So all these things you will be able to see when you observe their beautiful behaviors. Okay? Next. Then migration, whenever we hear the term migration, birds are the most studied subject. Okay? So birds perform different types of migration. Some travel only during the night to avoid predators. Some travel during the day because there will be big flocks, so they believe in safety in numbers. Then some travel non-stop. Like there is a bird called Varthur godwit, which goes to North America. From New Zealand and Australia, they start their journey. They come to Asia, then they go to Europe, and then to North America. Okay? Then from there, they breed there, and then when they take off and go to New Zealand and Australia back, they don't take the route that they came. They go through the other side. So there is no land in between, right? So before migration, they eat well and become fat. They take off. By the time they land, it takes about 9 to 11 days. By the time they land, they are slim and trim. Okay, these are some very interesting adaptations that they perform. They migrate that long distance without stopping anywhere for food or water or rest. They cannot stop because it is ocean in between. If they stop, they will directly fall and drown. Okay? Then, this is very important for you. When you start identifying the birds, how you need to document it. This format is the basic format that is followed across the globe. You can also use this format. So first you have to put serial numbers, otherwise every time you think of how many species you have got, you have to count 1, 2, 3, 4, 5, 6, which takes a lot of time. So please write down the serial number, then common name. Common name, like I said, most of the names are given based on the appearance, so please be familiar. If you don't know the common name, ask your parents or farmers or whoever nearby. You can ask them for the vernacular name, that is the local name. So it could be in Kannada, Telugu, Tamil, whatever language you speak. In that language, if they are able to give you the name, with that name you can easily find out the common name and the scientific name. Understood? So scientific name, don't worry about it in the field. Once you come back home, you can use internet or your libraries for some field guides, and you can write down the scientific name. Then the next is numbers. How many birds did you see? As in a particular species. So for example here, rose-ringed parakeet, how many I have seen?
9.
Very good. And why I use tally marks is, if I say, I don't see 9 of them standing in one place, right? I see maybe 1 or 2 initially. Then after some time I see another 2. Okay? So we use tally marks instead of writing the actual numbers. If we write the numbers, then we have to scratch and write again. So instead of that, we follow tally marks. Then finally the activity. What were they doing when you observed them? Some would be flying over, some would be calling while they are flying, some would be perched on a tree and then they will be calling. So these activities also you need to write down. And before you write all this down, there are few important things that you need to write down. First is the day, second is the start time and the end time. It is very important. What time you started your observation, what time you are ending your observation. Understood? Then the location and the weather condition. Because if it's a rainy day, you are doing your observation on a rainy day, the number of bird species could be considerably low. Okay? So if you don't write the weather condition, when you come and present your data, I will ask you on this particular day why you have found less species. You will not remember because our memory is not that good. Right? But if you have written the weather condition, you can definitely refer to your logbook and say, it was raining that day, which is why the number of species have gone down. Understood? For identification, I will give you some suggestions. One is, you can use lot of field guides, but whenever you are purchasing field guides, please remember to purchase a book that has the local species. Don't buy birds of Himalayas and search for birds of Bangalore in that book. You will never find it. Understood? So buy locally relevant books. If you don't want to buy a book, there are online databases also. Next. All these are different online sources that you can refer to help you identify the birds. And if you go to this particular link, there is a book which is Common Birds of Bangalore, which we have written. So it is available here for download. So you can download the PDF version and you can use that also. So 25 common birds that are seen in Bangalore are available with photographs in them. This PPT will be available later for your use, so you can take the link from there. Okay? Next. So here I will show you how to use one of the databases that I have just now showcased. This is iNaturalist. This is the most simplest of all those databases, and this can be used very easily. So when you go to your system or phone and enter iNaturalist and go to their website, you will end up in this page. Okay? So here there is an option called Explore. Click on Explore. When you click on Explore, you will get a page like this. Okay? This is giving you all the species of everywhere in the world. Do you need so much data? No. So what we will do, we are looking for birds, right? Here it says species and here location. So in species we will put birds and in location we will put Bangalore. Because whatever you are going to find in Varthur will be included in Bangalore list. So we are going to that list. So we can go to Bangalore. So now I have done that, and now it is restricted to Bangalore area and all the different species that are in Bangalore it is showing. Okay? Where it is showing, I have to scroll down. So if I scroll down, it is giving all these things. It will show observations. So the observations are in thousands. Because how many people, how many times they give the entry saying that I saw a crow today, they will enter. Tomorrow I see a parakeet, I will enter that. So all those are each observations. Okay? So we need to click on species. When we click on species, it will give you how many other species are there, only those species across Bangalore area what you have selected, it will give you the list. Understood? So you can scroll here and look for different photographs of and compare with what you have seen, and you can identify the bird. For example, I have seen something that looks like blackhead, but I am not sure. Okay? So I will click on this. So when I click on the particular photograph, it will open a page like this. It will show the trend of observation month-wise. In which month more numbers are observed, in which month less are observed, all that will be seen. But you don't need that data for now. Okay? You are looking to identify this, correct? So what you will do, there are so many images of the same species. Because not always you would have seen the bird sitting like this. Like I said, they won't pose for you. So you have to see all different photographs to understand and identify accurately if this is the bird that you have seen. And then when you scroll further down, see the geographical range also. See, if say for example I am in South America, I am standing in South America and I saw a bird that looks similar to black-capped. So now when I see the distribution here, its distribution is only here. So I am here. Can this be the bird that I have found? No, definitely. So this is a method that you can use to sort out or filter some of the species that is not required. Understood? Okay. Next, say for example, I still cannot make out because I am still in India and the range is also there, but I am not sure if this is the bird that I have seen. What I will do, I will scroll to this area called similar species. Click on that. Yeah. Next. So when I click on similar species, it will give me the birds, all the birds that look similar to black-capped. So at least in the initial page, you narrow down to what type of bird it was looking like, right? Then when you come here, it will give you all the other birds that look similar to black-capped. So from here, you can easily identify the species that you have seen. Easy? Same way it works in phone, only the orientation will be different. Okay? Yes. Next, we are all very smart people, right? But see how smart the crows are. This also happened in Japan only. You tell me why it did not happen in India. In Japan, crows eat walnuts. Dry fruits, you know walnuts, right? So walnuts are very difficult dry fruit to eat because the shell is very hard. You need to crack it and it is hidden in between the shell. How crows used to eat in Japan is they carry to a great height and drop it on the rocks. Okay, with repeated attempts, the shell would break and then they used to pick it and eat. Now you know what they are doing? They bring it to the traffic lights and wait for the lights to turn red. When it is red, they bring the walnuts and place it in the zebra crossing and then go back up and wait. When it is green, all these heavy vehicles pass on that, breaking the shells. Then again when it is red, they come down, eat it and go. They have given up on the hard work. They are now doing smart work. So looking at the crow, we also need to do smart work. Now why crows in India did not do this? We never stop in the line. So please follow traffic rules so that our crows also can innovate. Okay? Next. Now I'll give you a small case study which we have worked upon. So in 2016, Lake Conference happened in Muduvitra, the same place where it is happening this year. So when it happened there, in the college they had a disputed land. That is, this is a ladies' hostel and this was a football ground. Okay, so there used to be a lot of disputes. And when we heard it, we ecologists are opportunistic people. So when we heard it, we took advantage of it and we proposed to them to have a first ever open bird park in Asia for wild birds. Captive birds, open bird parks are available in Singapore and Malaysia and all those places. But in India or in entire Asia, for wild birds it is not there. So we proposed that, they liked the idea and they agreed to it. So we'll show you what we have done. So this is in March 2017 when we started the work. The land was like this. You can see the soil, right? It is very, very loose. Okay, and when we did a satellite image search, we found that there is no water body nearby, that is perennial. Seasonal water bodies are there, but not perennial. So what we did in the bird park, we decided to create a water body also for the birds. Next. So this is May 2017 when the monsoon had just started. The rain in Muduvitra is not like rain in Bangalore. If you go out during heavy rains, right, it will be like somebody is throwing stones at you. The water droplets are very heavy and it feels very painful. So with that intensity of rain, the soil here started eroding. So if we planted any saplings also, that would not survive here because with the rain that also get washed away. So we had to stabilize the soil first. Next. So what we did, we understand that grass stabilizes the soil faster. So we planted buffalo grass in the month of June. We are in the 1st of June, so a few years back. We planted this and 1 month later you can see how the texture of the soil has changed. Okay, next. So we had proposed bird park, right? So we had to observe the birds also. So initially at this stage we found only these particular species which are very common in any urban setup. Next. So we used the students there to plant the saplings. We collected the saplings which are local to that region. See, now the trend is plant native species of trees, right? So when you plant native trees, when you say native, we mean native to that particular region. don't consider entire India as native, because you cannot bring saplings from northeast and plant it here, or from Andaman and plant it here. They have to be in their own habitat to do that particular ecological role. So here we had accumulated saplings of that local region, and we used students to plant those saplings. And here you can see how the soil texture has completely changed, right? Next. So this is right after the planting. Few months down the line, see how the grass has grown very well, and it is holding the soil and preventing the erosion. Next. Same year, October. You can see the water is still silty, because there are still areas where the grass is not grown, and there are other areas from where the silt is still coming into the pond. That is why the pond water looks very silty. Okay, next. So here, as the grass grew and the saplings were planted, the number of bird species also increased. Lot of insectivore and frugivore birds started appearing there. Next. This is few months later again in December. You can see the entire water had seeped through. See, for any water body, when they talk about the water body, they'll explain further. The groundwater has to be recharged to the surface so that it can hold water in the surface here. Okay, so here, because it's a new water body, the water kept seeping in. Next. You can see in summer 2018, the water is not there at all, and the plants have survived though, and it is maintaining that green color. Next. So now fast forward to October again. Water is still there, and you can see all the plants are beautifully growing. And can you see blue boards in between? This is because every month I visit Arvas. Even now I am going there every month. So when I go there, all those students who were there at that time, I used to ask them, like, you planted this sapling, right? Tell me the name of the plant. And every month they used to forget the names. So now what they have done, they have put the boards by themselves near each sapling that they planted. Every time I ask, they just look at the board and answer me. See how we are asking questions is helping students to innovate also. Right? Now they are upgrading it. See, those students are passed out and gone. Now new students have come. They are new generation, right? So new technology they are using. They are putting QR codes near everything, and then you just scan and you will get the information about the entire sapling. Because here only names are there. But with QR code you can put a lot of information. Right? So they are doing that currently. So next. So 1 year later, we can see all these different species. Here it's a mix of all the different species, and it started looking like a healthy ecosystem from the birds angle. Next. So this is just a reference image for the size of the sapling. When we were planting, you saw that saplings are very small, right? In 1 year, they have grown taller than us. Okay, next. 1 year fast forward, October 2019. Can you see the water body? No, because the trees have grown taller. Okay, and it started looking like a very good garden. Next. 1 year again, fast forward. COVID happened and we could not go, but I was like very worried, saying that my baby I cannot go and see. So now you can see how it is looking. November there was a small gap in between COVID, right? So that time also we went there and we observed and documented the bird species there, and now you can see from a garden shape it is now shifted to forest. Next. Again 1 year later, the same area I am standing, but the entire area looks very much different, right? From what we started. Next. This is 2022. Next. See, this is last year in April, peak of summer. There is very good water in the pond. So 5 years we had told them that it will take 5 years for it to hold water. That much seepage has to happen. So exactly 5 years later now, water is holding there even in summer months, and it has become a perennial pond. Okay, and you can see that same sapling how tall it has grown. I can't even cover it with my camera. Okay, next. So that concludes my presentation for today. If you have any questions, you can ask. Otherwise, we can move to the next session. Any questions? Yeah. Good question. You tell me, if you also have to get a shock, what is required? Electricity is required, but in those wires. Yeah. And? See, when birds are standing on those electric wires, do they stand on one wire or multiple wires like this? If they by mistake touch the other wire, they will be electrocuted. Now you understand why?
How can I prevent it, sir?
They have to be careful. And in some areas, they insulate the wire also, so that even if they touch, nothing happens, but not everywhere. Good question. Nobody knows this. We only know the lifespan of captive birds because we can study them. Being in captivity, we can observe from the time it hatches till the time it dies. But then in the wild, we can definitely not observe that because we cannot fly behind them and keep observing them. So it is not known. But general tendency is smaller the bird, smaller the shorter the lifespan, larger the bird, longer the lifespan. There are birds that live only for about 6 years. There are birds that live for 30-40 years also.
Ostrich is a very big bird,
Yeah.
so it will have more lifespan.
Yeah, it will have a longer lifespan compared to the smaller birds. And coming to mapping, so now we have a dedicated session for mapping, so you can learn how you saw all those satellite images and maps, right? So using Google Earth, how you can create your own maps for your study area. Whatever study area you choose for your project, that study area map is a must in your project. Okay? So how that can be done, Tulika ma'am is going to explain that for all of you. Please observe carefully because she is going to show a lot of things like how I showed IISC restraint. So like that step by step she is going to show. Please write down all the steps so that you can easily do your maps. Okay? Are you all ready?
Yes, sir.
Okay. Over to you. Okay, all of you stand up. Keep your books on the chair. Okay. So when I raise my hand like this, I want to hear one clap, but all of you should clap. Understood? How many did you hear? 5, yeah. So many.
Take a deep breath.
I didn't raise my hand.
1, 2, 3.
Okay. My hand is visible, right, to all of you?
Yes, sir.
Okay. I said this hand, no? So you need to focus. Close. Yes. Okay, sit down.
थोड़ा सा ma'am. Everyone's phone is there? Yes, ma'am. We all know how to use Google Maps? Yes, ma'am. We know how to pinpoint our home? Yes, ma'am. How to go from school to home? Yes, ma'am. Very good. So, using that technology only, we map whatever plants or birds you have identified. Okay? So first, I will mention about two apps that you can install in your phone that will make your life easy. First one is GPS Map Camera. Better. GPS Map Camera. Yes. Then, okay, ma'am. So this app looks like this. When you go to Play Store, it will look like this. When you install it and take a photo using this map camera, you will get a photo similar to what you get from camera. The extra thing is you will get this thing, which will show you the details of your location, the date, and also the time. So when you are in the field, looking at a tree, okay, this is the ficus tree. This is the, when you take a photo, then you have the location, date, and time. You can use it as a logbook. Write it down. Will it be helpful? Yes, ma'am. You can use it in your project? Yes, ma'am. Okay. I will show you one video. I saw this peacock. Can you see all of you? Yes, ma'am. Where I have located, can you see? Yes, ma'am. So just using the photo only, we also can see the location. Right? Then, this is a water bird. You can see it's flying and gliding down like an aeroplane into the water, right? Where I have taken it? Date and time you can see? Yes, ma'am. So you can use it for your field works, right? Yes. So that you can capture what is happening in your field. Okay? So you have to remember this. Lat and log. This will be required afterwards. Okay? Yes. Latitude and longitude. Very good. How many of you know lat and long means? Very good. So then we move to the next app. That is called Google Earth. This comes like a football, blue colored football. Okay. So if you install this app, similar to Google Maps, you can see this blue colored dot. This one. Yes, ma'am. When you click on this app dot, what happens in Google Map? Wherever we are, it circles back to that particular point. So it works in the same way. You have to click on that point, and that point you can save in your phone. Okay? The better part, or you can say the advantage of using this application is you can add a description or the, we can say, the different data that you want to associate with that point. Okay? Suppose this point I have located. Okay, now the climate is cloudy. I can't find my friend, and there is a bird nearby. Like that, all the descriptions you can write. That is the advantage of this app. Okay, so two apps I have shown. What are those two apps? GPS. Very good. So now we will learn how to create maps. Like sir was showing now. Those look very good. We will also make and show to sir. So we will also send it to us. So how many of you know about Google? Okay. Who do not know about Google? Okay. So who do not know? Yes. Can you tell me what it can be? Yes. Just guess. It is linked to Google Maps that is used every day.
Can search the places.
Yes.
Places. Nearby places that we want to go but we don't know where it is. And
Any more points? Anyone want to add? No. You know Google Earth. I want to know from who do not know. Anyone? Yes. You. because this is scientific. This will be scientifically prepared maps. Okay. Then the interesting thing about Google Earth is it also acts as a time machine. What does time machine do? Yes, very good. So using Google Earth, you can go back to time and see what was happening. Suppose you know Varthur Lake is there nearby. So if you go back, you can see what was Varthur Lake like 10 years ago. And what is now. So that is interesting, and that will help us also to identify the changes. Very good. Then we will install it. If it is not in our computer, then we need to install it. For that, simply go to Google and search for Google Earth Pro. What you have to search for? Google Earth Pro. Very good. Then we will get this link to Earth versions, and we can click on this link. Then we will be taken to this website. This looks like this black website, where if you scroll down, you can see this box to download. You click on this box. Download Google Earth Pro on desktop. Simple.
Yes, ma'am.
Not complicated. Not okay. Now we have downloaded in our desktop. Now they will ask for some permission. You allow it. Then follow the steps. It will be, the installation will be completed. When the installation will be completed, it will look like this gray football on your screen. Okay. Now the installation is completed.
Yes, ma'am.
Okay. So which what we have to search for in the Google? Very good. Then we have to click on download Google Earth. Then you have to follow the process and it will be installed. Okay. पूरा complicated हो गया? इसमें yes था? So if we click on this Google Earth Pro, first you will get a glimpse of Google Earth. Then you will be taken to your current location, like this, like you are traveling in a spacecraft. Okay. So now I will give you a small tour of Google Earth. Okay. What are the things there? First thing is, on the top left corner there is a menu bar. Then just beside menu bar there is toolbar. Then just below the menu bar there is a search bar where you can search for the location, and I have searched for Indian Institute of Science, and if we click on that location you will be taken to that location. It is simple as Google Maps. Then just below that the points, lines and polygons that you add, that will be listed in your places column, and there are multiple base layers available. Okay, so what are the things in Google Earth Pro? Menu bar, toolbar, search bar, list of your pages, base layers. Very good. So now you tell me what is point, what is line and what is polygon. Point means a dot. Point is a line. Line is a red. Okay, point is a dot, line is a line, polygon is a polygon. Line is a Line which does not end. Okay. Suppose I am seeing a river. If it starts from a point, it ends at some time. Will we consider it as a line?
Ma'am, lines are like roads, rivers and roads.
Okay.
Points
Points
are
are
places. Same. Polygon is the area that you can mark.
Okay. So points, what we can show once again. Certain places. Right. Then trees, what we'll use? Points, lines or polygons? Points. Very good. For roads, lines. For railway, points. Lines. Okay. For our house, polygon or point. If your house is very big, you can use polygon. If it is very small, like a sacred grove or like a temple or something you want to mark, polygon. Point, polygon, all can. So suppose you want to specify a location, then point. Here have made the whole campus as a polygon. Now you can see the difference between all the three. Yes, ma'am. Clear now? So now we learn how to make all the three. Okay. First one is point. So to identify the point, you remember that lat long we had while we were taking the photo. Latitude and longitudes. Here, if you click on this yellow thing, can you see this yellow thing? Yes. This is called add place mark. What is it called? Add place mark. Right now. Yellow pinpoint thing. That is called add place mark. If you click on this screen will come. Here you can see this latitude and longitude. Here you can put those latitude longitude values where you are taking the photo. If you do not know the location. Okay, first scenario. Second scenario, if you know the location, so you know your school's location, you can drag the point and put it on your school. Then it will, this latitude longitude will be automatically updated. Okay, two scenarios. If you know the latitude longitude, and even when we do not know the latitude longitude. Okay, complicated. No. Okay. So here what I have tried to do, I have identified the main building of our campus. Okay. So here I have changed the name. I have changed it to IISC main campus. You can see now. Then latitude longitude it is already given. Then the symbol. Here you can change the symbol to this one. This is the appropriate symbol for among all the symbols. And this latitude longitude you can also note down in your notebook, which you can refer to in your project report. Okay? Because finally we have to prepare a project report to submit. Now, for that we need this documentation and all. Correct or not? Yes. Then one final thing that we need to do, from this style and color option, we can change the color of the point. Okay? And finally you have to click on OK. Otherwise this won't save. So please remember to click on OK. If you click on cancel or outside, it will be gone. Okay? So tell me the steps how to create a point. Latitude, longitude, change symbol, and okay. So here this is the symbol. It looks like this black dot in a black circle. Okay. Now this is the Varthur lake. Okay. In Varthur lake, suppose you have gone for water sample. To water sample, there are 3 points you have to take the samples for. Now, Gagana ma'am will explain where to take the samples for. Suppose this is the inlet. Here I have taken the sample. Now, how will I know what is the location? For that, when you take the photo, you will get the latitude longitude. When you come back to your school, you can give the latitude longitude in the grey box, and it will automatically come to this location. So like that, you can do for the outlet locations or the other 2 locations also. Okay? You understood? These are some examples of macro files that you can see like floated on the top of the lake. Have you seen? Yes, ma'am. These are very common, right? So suppose you are marking some of these plants on near the water bodies. These are all water bodies plants, right? So that also you can mark the locations. Correct? Yes. Okay. So I have a quest for you. Your first quest will be identifying your school and also your house. That you all can do? Yes, ma'am. Identified in Google Earth Pro? Yes, ma'am. I have another quest. You can say sub quest. That is TVRAMACHANDRA in his presentation he was mentioning about hugging a tree. How many of you remember? Hugging a tree. Right? Measurement. When you feel low, you go and hug a tree. He mentioned it, right? So there I heard around 550 trees in your campus. Yes, ma'am. So when you not interested in hugging trees. Okay, there are many trees in your campus. You can hug a tree and you can take photo of the tree so that the latitude and longitude will be there. So you can come back and note down the latitude longitude in Google Earth Pro, so you will know which tree you have got. Right? Yes. Anyway, you can do. Okay, outside also you can do. Okay. Next is how to make a line. Here I have tried to identify a line that goes behind our campus. So this I have done using this add path option. Okay, that is with circles and lines. This add path option, when we click, we can digitize the line and finally give the name and also change the color. Okay? Okay? Yes. Very simple, there is no complications. Now, how to measure? What is the length of the road? To do that, there is a called measurement section is there. Style and color will give you the color. Then just beside that measurement section is there. Here you can check the length also. While you check the length, you put that in your notebook also. And this you can do it in different units as per your requirement. Okay? So what are the steps? Okay, you have to press, otherwise it will be gone. Okay? So I have a example of line data that shows the drainage network of the Varthur catchment. That Varthur catchment delineation parcel will show you. But the length of this catchment area, I don't. You can see the drainage network. The length I do not know. You can calculate this, right? Using Google Earth Pro, using measurement tool. So you will this also gives you an idea for your project. Okay? So I have a second quest for you. You have already marked your house and your school, right? Now there will be different routes from everyone's house to your school, correct? So now you can mark the road and you can see the distance. If you have to walk, how many kilometers you have to walk. Okay? All agreed? Yes, ma'am. Very good. So now third one is how to make a polygon. So polygon will show us the area, right? So here I have tried to identify the whole campus using this add polygon feature. Which feature? Add polygon feature. Then I have given the name, same color and the area, both the color. This lines mean the boundary. See, I have given the pink color to the boundary, so it will be the very and this is the green color for the area. Okay. Now same like we have done for the line measurement, we can do for area also. But what color to give in that polygon? There are some universally accepted colors. So suppose I have given water body red color. She has given water body as blue color. He has given water body as yellow color. Now when we have all came together and put our maps one after just beside one after another, now we are confused what these different colors mean. So to remove that confusion, all the big heads of the world sat together and decided, okay, we'll use these conventional colors for these land use categories. Okay? So for vegetated areas we use generally green color. Which color? Green color. Water bodies we use blue color. Agricultural land we use yellow color. And paved areas or built up we use red color. Okay, again green we use forest. Then yellow you use water. Red agricultural. Okay, again These two you can note down in your field book, and this for this also you can use different units of measurement. Is that okay? Yes, ma'am. Now, can you tell me in this map which areas I will take for the polygon data? Okay. Only Varthur Lake. All so the Alsur Lake, Chalagata Lake, Balandur Lake, Varthur Lake, all we can take for polygon data. Correct? But I do not know what area does this lake cover. Can you do that? Very good. So you can do that as your project also. Right? This is also a suggestion. Then coming on to your third question. Okay? First and second question we have done now. This is the third question. That nearby Varthur Lake is there. So you can all digitize the boundary of Varthur Lake and find out what is the area. Yes, ma'am. You all can do that now. So agreed on request. Yes, ma'am. First one is point. Okay, third one is okay. So now coming on to our time machine view, where we can see the past images. So that we can use using this. You can see this clock type thing. Let's zoom in. Now you can see this clock going backward. So this means going to the past. So using this, if we click on this thing will come on your screen. This will come in very chota here. So I have zoomed it and showed you. So here you can see from 1980s to current time, it will show you the images. Okay. So this is the IISC campus or International Science Campus. This is the scenario in 2011 and this is in 2023. Can you see the changes? Yes, ma'am. So you know what is the importance of this change detection thing? Now if we see the Varthur Lake, this is 2023 image. Okay? You all have seen? Yes. Okay. Now going back, this is 22. Can you see the change? Yes, ma'am. Then again going back, 2021. Can you see the change? Yes, ma'am. Again I'll go back again. This is 2011. Can you see the water? Yes, ma'am. There was no water in 2021. But we can see water this time. Correct? So from this you can see the change analysis. You can do that? Yes, ma'am. Okay? So now we have created the points, we have created the lines and we have created the volumes. Right? Now it is time to save the map. So we need to prepare the project. This is important now. So for that, this option is there which is called save image. What is it called? Save image. Okay. If you click on this, everything will come. Here, this thing will come where you can give the title and also the description. Okay? Then this corner, the region will come where you can identify what this line means, what this polygon means. Okay? Then using this save image option, you can save the map. Okay? Agreed? Yes, ma'am. Now the final output will look like this. Any confusion? No, ma'am. Okay. So to create this map, we have to keep in mind 5 things. How many things? 5 things. 5 things. So first one is we have to give a title of the map, which will show us what is in the map, what this map shows us. Okay? The second thing is the legend, where it will show us what are the things in the map. Okay? Third thing is the north arrow, that is here. I have shown like zoom version. From back you can see. Okay. So this north arrow, which means which way is the north. So suppose if I have tilted it, like rotated it ulta, then this north arrow will go like this. Correct? So if anyone has tilted the map, we can see the north arrow and we can see, okay, this much this map has been tilted. Okay? Then the fourth one is the scale that shows that is here. Okay? This will show how much area, how much for our further research, right? For that we have to save it. Okay? To save it, what we will do? See, I have taken multiple tree points. I will right click on this. Okay? If I right click, this thing will come. Different options. Now this you have to click on this save place as. Save place as. Okay? If you click on this, then it will take you to one of your location in your system where you can give the file name, and you have to be very careful and save the type as .kml. What type? .kml. Okay, so what is the steps to save the map? Right click. What is the file type? What is the file type? What is the file type? Again, what is the file type? You won't forget, na? And now you can use this whenever you want it. Even after 10 years after, you will have this. Then also you can open it and use it, right? Now the main thing is to document the research. Whatever you have done, you have to document it, right? Now we can do it. There are 4 major steps in your documentation. First is exploring your library, or whatever the content is there already on your focused point. If you are interested in water bodies or soil or biodiversity, you have to go to library, see what are the contents available, explore your library. Then step 2 is you identify what are the methods to do your research. Okay, so you can plan it like first you have to plan what are the things you have to do. Then based on those things you have to collect the information, and third thing is you have to analyze. Okay, based on the information that you have collected. Then the third one is writing the report. Writing will require 4 stages. First one will be your introduction. Second one will be materials and methodology. Third one will be research, discussion and conclusion. Okay. Now we have done all our project. Everything is done, but we have done it all by ourselves. We do not know how much it is correct and how much it is wrong. For that we have to come to a teacher or some experts who will give us some feedbacks. Okay, this thing you can do it like this, so it will be better. So now you can go back and do the modification and then come back again. So this will help you in modifying and to make the project better. Okay. So what are the 4 steps? First one. Okay. Now we'll continue with the research. Agreed?
Yes, ma'am.
All agreed?
Yes, ma'am.
I can't hear the last.
Yes.
Yes, ma'am. Okay, very good. Thank you. Are there any questions? No. All clear now?
Yes,
Mandi? Okay. We'll go for lunch. Yes, ma'am. Thank you.
We know what is point line for you and how to locate and how to locate a particular location or how to map that the route and that study area to delineate study area. So now we will know that as Vivekanand told you that I will tell you that how to map the catchment area, especially this Varthur catchment. So what is catchment?
Particular area.
Catchment of watershed you have heard earlier.
Perfect area.
Any points of can be a point. Mark can be a point. And if you are delineating or mapping a route, then it will be a polygon. And if you are outlining a specific area, so it will be polygon. So nobody you have heard about the waterside and catchment. So basically waterside and catchment or catchment, what is that? The high area which shed water to downstream. Means it is a boundary of all the most elevated area in the region. Suppose we have 4 or 5 rivers. So the boundary, the line joining the topmost portion of that highest elevation of that land surface feature. Land surface means whatever land features are there. So the top boundary which sheds water down in the streams or in the lower lakes, that is called the entire area is called the watershed or catchment. So we will know how we can map this using remote sensing data and GIS data. Remote sensing data as remote sensing, what we acquire remotely means we are sitting here and we have launched satellite. Satellite will capture the data and we will download it. We will get it from there and we will use it for means we won't be in the direct contact or we are not taking the primary data. Means we are not going to field to capture the photo and coming back then we are doing. It is a remote thing. Means satellite are doing for us. We are just getting it, downloading it and we are analyzing it. So it is a cost effective method also means mapping area catchment area by going in the field. It can be costlier and labor expensive also. So we will analyze this remote sensing data. So what we need is for remote we need this DM. DM means digital elevation model. This is a kind of 3D structure of our Earth surface. Means if you suppose if you close your eye, one eye, if you means how can you tell that this is of 1 meter kind of thing, this height, this is 2 meter. No, by visual interpretation. No, just like a prediction means or a certain thing is to this much mark. Means you have a kind of depth perception. Depth perception means something is locating somewhere by two observation point. Like we have two eyes. So if we close one eye, we can't means we are unable to identify the height. Suppose if you see closing your one eye, your finger, you will see like a 2D surface. Means a plane. If you can try also. Like if you close one eye and you see the world or around you, you will see a plane. Means you can't see that some in a 3D. Means which have a volume. So for that we need two observation points. So for that this satellite will click an image at this moment and the another image at the right moment next to that. So we will have two images of same location. Then we have the same location and now these are 2D images. Means what we captured that is there are images. If we get print of that image from what we have captured photo, it is a 2D. So we will overlap those two images which are of the same area. So it will that is called that will give a depth perception. Means that will give you a perception that elevated land or that mountains are highly elevated. These water bodies are as a sink or deep down. So we will use this. This is called raster data. Means in that we have raster and vector data. Vector data, what Tulikaman told you that point and polygon, that is made up of pixels. This one, if you zoom in, if you have captured your image and you zoom in too much, then you will see square shape pixel. That is the unit of that image. That is called pixel. So that is called the raster image. If you zoom in draw on a line or kind of thing, you won't be able to see that pixels. That is vector data and this is raster data. What you can at the end, what you have the unit value as a pixel or a square. So we will need both vector and this. Then as a raster data, say file, this is the boundary file or boundary file as a vector data. This boundary is of Bangalore urban district, and this is we have clipped it for Bangalore urban. This is the value, means in this what you can visualize. What you can visualize, these darker areas are the sinks or the low-lying areas, means river valleys and all the water bodies. And as the gradient increases to white, that means that elevation is increasing. Means these are the high land or elevated land. These are the elevated land. So we can't means visualize properly from this input image. So we can provide a color map suitable to our visualization. You can provide a understandable color map, so you can visualize this properly. So this the deepest area are shown by dark blue. Then as the elevation increases, that increases to light blue kind of thing. And in the land, in the surface, that green is turning to red as the elevation increases. So with means of this data, we will try to map our catchment area. So now where to get this data? Means this raster image. Means this input image we will get it on this Earth Explorer, NASA Earth Explorer data portal for free. This is for free, so we can use it. We will type Earth Explorer in Google and it will redirect to it will suggest this link. So we have to log in or sign up for this portal. So we will get this kind of interface. So we will highlight now we will highlight our study area, means where we want to do our study. So we will highlight that portion, and based on that, if we will search, and now the second comes the data set. Means this is the portal. This we have prioritized our area. Then it comes the data set. So in data set, it is not here, but SRTM data. You will note down, because it will be useful for you to delineate your catchment area or the Varthur catchment. So you will use SRTM data, which is 1 arc second. This is 1 arc second, but we use means that is of 30 meters ground resolution. 30 meters ground resolution means we can distinguish between two objects which are kept apart 30 meters in the ground. That satellite can differentiate between two objects which are clearly differentiate, means they can identify these are two different features which are kept 30 meters apart. So we select that criteria. Then we click the data set. Under the data set, in digital elevation, we will get this SRTM, and SRTM 1 arc second globe we have to opt for that. So it is highlighted also. So in after that we will get because we have prioritized our area, so we will get the series. Means that raster image. This is one raster image. This is the second one. So these are two different. So it is coordinates, and this is the location path of road. So these are two different images, or we can say DAM data, digital elevation model data. So we will download it. Here that download button is there. So we will download it, and it will ask us for the which data version we want to use. So we will use this GeoTip. GeoTip means it is geo-rectified. Means if you download it, like if you import an image in any of the Google Earth engine, it doesn't have coordinate system. Means GPS. As Tulika ma'am told you that capture image, that is extensively for that app. That app is giving you that service that it can locate that lat and long. Otherwise, if you capture from your simple mobile, you can't get that lat long for that image. So GeoTip images means they will have their coordinate values. Means where all means x and y latitude and longitude. So we have to opt for this GeoTip version. Now we will use QGIS. QGIS is an open source. QGIS is an open source GIS software in which we can do our mapping and all. Means it is free of cost. So we can use this for our GIS work, remote sensing, data analysis work. So we can have a greater visualization thing. So in that, if we want to add that now that is in our repository, means in our local disk. So we want to get it in our this QGIS. So we will go to layer. It is the main layout. This will be the main layout of the QGIS. So we will go to the layer. Then we will add layer. Then add layer. You can see that different options are there, like raster, vector layer, raster layer. So this is what we have downloaded is a raster layer. This that are made up of pixel and that has the XY value. So we will opt for this. We will click add raster layer. So after that it will look like this. Raster layer. This pop-up, this dialog box will open, and you have to just click that add button. So it will get added to your GIS interface. So you can see it will be added like this. These are the layer panel and this is the display panel in QGIS. So these two, because our Bangalore urban is not covered in a single DM image, so we downloaded and we will use two different images. So these are the two things. By the time when you what is catchment?
Water side.
Water side is this I didn't keep it this way. Anyone else?
How many of you know about GIS? What is the full form of GIS? Huh? Raise your hands, everyone. No. So, GIS, yeah, please explain.
So GIS is a platform in which we collect data, we store data, we map it and we display it, and then we can publicize it also and we can share it. So for that we have a specific
GIS means Geographical Interface System.
geographical information system.
So how do you interface or get the information? Because all the satellites are there. So from there, any image are there. this is captured only data information. Then suppose you are seeing me, I am changing, you are looking at your friend. Yes, how? These all are interface. Okay? Are you getting? Yeah. What you want to code? Yeah, please. What you want to
So now we have added that those two images in our GIS software. That is QGIS. So now we have to that means we have to merge this. We have to merge these two things because these two are separate, and our study area lies in between this. So only one image is not enough to visualize, to cover our study area. So we will have to mosaic it. Mosaic. Now we are mosaicing two different raster layers. So these are two separate images, and we are making this as a single image. So we will click at raster. In that this image is there. This is at taskbar. We will get it at the top of the QGIS. Then in this area we will call merge. This tool will do. It is kind of join, but in geographic terms, joint means a different terminology. So we will use merge for raster layer. Join is used for vector layer to join two tables or two things. So we will not use join word. We will use merge or mosaic image. So there, if you click that, so we will have this interface, and we will choose from this three button. We will choose an input layer as those two layer, and we will give the location where we want to save it, and we will name it as like what I have given. So now we are locating it, you are saving it in your personal space. So based on these two images, we will make it as one image. And then we have to run it. Now it has created a single layer. As you see, there is no this joint in between that. So because that line was because of that two different things. Now it has become a single thing. So now about that polygon of study area, from where we will get it. So for Karnataka specifically, we have this KKEHS program, village level, that vector data in the boundary of state. You will get it, or another new source are this water resources institute and this river area. So from here also you can get that outline of study area. Now we will add that vector layer in our QGIS. Earlier we showed you about how to do analysis in Google Earth. So this is we are doing in QGIS. So for that, as the same, we did for adding that raster layer or billion input layer. We will add this. We will click layer, then add layer. Now we will opt for this vector layer instead of the raster layer. This we will add as a vector layer. So it will open the similar interface as like earlier. Earlier it was highlighting that raster thing. Now it is highlighting that vector thing, and we have to it will display here in this box. Now we have to simply add this layer in our display panel. So This will look like this. This will look like this. This is the boundary, outer boundary of what BBMP means. This is BBMP Bangalore. So that you know that we have merged that these are the two things. We have merged that in a single, and now we have added that this shape here means we are this entire thing. We don't want to enlight our study. We only specific to our study area, which is BBMP Bangalore. So now we are specific to our study area. So we have to click that raster here. Means we have to click that specific image for our study area. Our study area is BBMP. So from that master, from that what we have modeled into here, we will click our study area. That we can do. So that is raster. So we will again we will go to the raster, and like we went for this and here, we will go now to this extraction. In extraction, we will use click raster by mask. Click raster by mask means we have a boundary and we want to mask the raster layer. We want to click a raster layer. So this will display like this. So in input, what we will do? We will give that DM layer, the raster layer, and we will give a target CRS. CRS means coordinate reference system. Coordinate reference system means what is the system? Reference system we are taking to locate a particular location in the world. So as you know that our Earth is an elliptical, so we can't project it in a plain paper. So we have hypothetically we have assumed that if we stretch that our Earth, spherical Earth, so it will look like Earth in a 2D plane. It will look like that. So we will use that approximate coordinate system for our study area. And we will assign that other values outside that boundary. We won't need that one. So we will keep it as a 0. So with that we will we will give where we want to save this location. This is the output location, and we will run this integration. So as we run this will become this. We will get it for the specific BBMP boundary. Earlier it was full 2 sheets because it was a too large covering a very large space. So we are interested in our study area. So we will get it after that clipping that from the that layer. So now we have to delineate water set for our specific region. Suppose we have Varthur region or Varthur lake catchment like that. So what we will do? We have to first reproject that DM. That DM is in global reference system. So we have to specify. We have to project it in our local Indian or means local reference system. So we will reproject it first. I will show you step by step. That we have to fill things. Means that raster layer what we downloaded from web or from IISC, it doesn't have that discrepancy. Discrepancy means if there is something null value, means no data for some pixel, it will give no data. So we will adjust that from this fill thing. So then we will define based on that raster value, we will define the flow direction. Means how the flow of these streams are there. Then stream order means what is the order of this stream. Or stream order means if it is coming from a healer direct range, it is coming, then it is a first order. If it is joining in the downstream with a bigger river, then it will become second order stream. Then again, if the same second order, second order means two big rivers are there. If they join, then it will create third order stream. That will be a bigger river or bigger stream as compared to the earlier one. So this is the stream order. Stream order means the which are newly or young rivers. That will be ordered at 1, and it will be greater higher as it becomes larger and larger. And after that, based on that stream network, we will delineate our catchment area or basin. Basin, catchment, and water set. These three are the similar words. So first we will reproject our what we have clipped our dam. We will reproject in our local reference system. So again, we will go to the raster. Again, we have to go to the projection. We have to project it in a 2D plane. That 3D model of Earth, we have to project it in a 2D plane. So we will opt for this ref projection. So it will we will get this interface. In this, we will give that what we have clipped that layer for our study area. That we will give now input, and we will specify our target, means what we want that local means reference system. Then we will use this resampling technique. Means resampling technique is the nearest neighbor. It works upon the neighboring pixels. Based on that neighboring pixel, it will identify what is the probable, means based on the, if there is some discrepancy in the, means some null data values will be there, it will be consolidated by the nearby pixel. So we will give the no data value. We don't want out of that study area, so we will put 0, and we will give the output location. So we will get a image like this, and now we have to fill the things. Means what all no data values are there, we have to fill it, and we have to make a consistent raster, means consistent raster layer which doesn't have discrepancies. So it will help in our analysis, means catchment delineation. So we will go to process. This is also available in the toolbar. So we will go to processing toolbar, then toolbar. Then processing toolbar, we have to simply search here field sync. Then it will suggest, based on that, it will suggest field sync. This Wang and Liu, they are the author, means they derive this method. So this method we have to choose, and as we choose this method, it will display interface like that. So now we have to input the image what we have reprojected. Means what we have reprojected for our local study area. So now we are using that latest raster what we have processed. So we are using, earlier we have used that merge thing. Now we are using that reprojecting to fill the thing. So we will specify our output location, and based on that we will run this one, and we will get an output like this. Now in this image, these are the null data values. You can see.
Yes, sir.
Now we have filled that thing. It has become a smooth raster. Earlier it was like this. So to overcome these null values, these are the null values in that raster. So to overcome from these, we do this fill sync. So now we have to delineate the means the stream or the stream network for the study area. So we will choose. We will again go to the processing toolbar, and there we will search for channel network. So as we search for channel network, it will highlight at that this toolbar, and we will Again, interface will open, and we will give now the what we have done the previous step, that fill them means that fill then we will use that, and threshold, what this threshold it means what order of stream you want in that network area. Means if you want all that small streams also, means that will increase your computational time, then you will put 1. Otherwise, if you want higher order stream, you can put this 5 or 4, 3, whatever that study, however small or bigger your study area, that depends upon this threshold. So we will specify the flow direction, and we will select, we want to select this flow connectivity, we don't need it, and then we will select the standard order. Standard order is that order of the stream. It will be the order of the stream, and based on that we need channels. So we will highlight that channel. These are the checkbox. Means the thing we need, we will check it. Otherwise we won't check it. So we will now run it. So it will, we will get an output like this. Means these are the streams. All blue, you are what you can see. These are the streams, and this is the direction. Means in which direction? Means these colors are visualized based on the 8 direction. 8 means primary 4 directions are there, but 8 based on 8 direction. The direction of flow, it will be highlighted by this method. So if we give higher order, we will get for BPMP this kind of clear means big stream, major stream. So and for under that basis, what we will get these bases. These polygons are the different bases, and these are the main water basins or catchment or watershed, what you can say. These are those basins. Here, this is the KC value in which Varthur is in KC value. This side Varthur is here. So this is KC value, this is Uswavati, and this is our bar. So what we get from that channel and network analysis, we got all these streams, and we got means these streams means this entire area is setting water in this particular big stream. Means for the smaller region, these are the streams. Means these are setting water in these areas like this, and finally they are accumulating that water in the main stream, that bigger stream. If you want to select those specific, now so this black and this thing is that what we got from that last layer, what we generated that standard order thing. So this dark white and the bigger, this smaller light white shows the lower order stream. This bigger shows the higher order stream. So if you visualize, this is Varthur area, and this is this parallel line shows that lake area. Means this if the line is parallel, means there is a equal leveling kind of thing. That means our water body is there. So this is the water body here, and we will draw now. This is earlier what we got, that is automatically based on the stream network. But we have put the threshold. Based on that, it automatically generated basin. Now, if you want a specific catchment area, watershed area for your region or for your study area, so what you have to do, you should know your means outlet point. Outlet point of your drainage means that catchment area or water body means the outlet is that where the all the water is draining out in the at the final. So in so we have to specify a particular specific location that is output. Based on that output, it will generate the catchment area. Means that area will set water in that. So we have to specify either that specific point. So we will click that raster layer. So it will display this much this box, and it will show the information based on that. So we will take this x coordinate and y coordinate. This y coordinate. X and y will give the particular location of that point, outlet point. So then now we got that specific outlet point at location of that point. Now we will search in that same processing toolbar. We will search for upslope area. So this upslope area will delineate the catchment area based on outlet point. So earlier what we use channel network, which generate the water set or catchment area based on the river network in a broader thing. So now we will if we click this thing, we will get an interface like this. Here in target x coordinate, we will put that our that location x value coordinate of our outlet point. Here y, and here we will use that what we have filled TM what we processed during our study, and we will use this deterministic aid. This will be technical, so I will give not focus on this. So you have to choose for the deterministic aid. Aid means the direction. Aid means the aid direction. Based on that, it works on the base of the neighborhood principle. So it will give an output like this. So this is the Varthur this is the Varthur lake. So this is the downstream, and based on that, what all the stream, small stream, bigger stream are shedding water in that particular lake. You can visualize like this, and this is the here Varthur lake have two outlets, one on the northeast and one on the southeast. So based on that, from where you want to delineate that or the outlet point what you consider, based on that catchment area will be delineated. So this is the final for Varthur area. Thank you.
In computer, we make a duplicate images.
Duplicate images.
Yeah. This is technology.
If we texted, they'll give us information about that particular thing.
Give you answer. Question and answer.
Can I make?
We can do it with photo also. We can.
We can make songs or.
Okay, so I am from IISC and I have created some application. I will tell you what it is first. So this one is Varthur Lake Information System. As you can see, these are all the names.
Paras has explained. These are the professional services, the ecosystem services. How much the lake generates? How much is it worth? So as you can see, this is the biodiversity after that. So it has microalgae, zooplankton, ichthyofauna, microphytes, and lastly the water quality parameters, which Ravina will explain. With this, you can also see the encroachment. What all encroachment has done in Bangalore lakes. So if we go for other lake, you can see how much the lake area is, how much is enclosed. And what is the documented documents available? This is the area in the red. This has been enclosed. Okay. Agara lake. So this is another application. This shows the land use, land cover, and all the other parameters which Bangalore is facing right now. This Bangalore urban information system. So which ma'am has already explained. Red means yellow, agricultural, others open land, everything, and green is for vegetation. How the build-up has changed. You see, the red color is increasing little bit. This is the build-up. The red is showing the build-up. So build-up is increasing in Bangalore. This is fully constructed. Industry, green and agricultural was there, obviously. And the Denver, this is the colored pipe which Paras is explained. So this you can see the lowest height 739. Where is it? Lowest area, a little bit over there. And the dark brownish, blackish color, where is it? This is the IISC point. The IISC gate is the highest elevation point in Bangalore. Yes. So this is the colored part. We will explain how you construct this. Okay. And go for lakes. These are the lakes. And you remember the channel you was explaining? What is the channel? What is the channel which Paras explained just now? Channel. Now, what is it? Streams. So it is how your lakes are connected. Varthur lake connected to Telugu lake. These are all streams. He has mapped it on the DA elevation model. On this, how it is associated with the lakes. Understood all? To be a little interactive, I don't stay
explaining of session. If he like fell off, his name is something. Let's say Ram. Is it useful to me? It's an information. But the information that is useful to me, your names, what are you going to do, your projects, those are data. The information that can be useful to anyone is data. Okay. So once again, imagine any tiny piece of information.
My date of birth.
Your date of birth. Okay. So with your date of birth, we can analyze how.
What are you?
Can you
What's your name?
What your DDS time is that you are given. So your body also collects information. Right? You're running, you're seeing anything. Yes. But it's just information. But if your brain has to move somewhere, there's an object coming to you. If someone is punching you, you have to move, right? That information becomes data when you have to, the brain has to analyze it. He has to work on something. So data in our daily life. Any one example?
Studies about studies are mobile data.
You guys are fitness trackers. Yeah, it collects your steps, your heart rate, oxygen rate. What happens to the blood load?
How much calories we have burned in our body?
Let's say your Amazon account, Flipkart account, they'll keep reminding you again to buy it again. There's something left in your card, right? Yeah. Everything is just data. Now getting to intelligence part. The areas of intelligence. How you can show that your friend has some intelligence. What are the 4 main criteria?
The behavior of
Your
his
friends have 4 of them or what? 3. Nice. So you think this 4 main concepts, you use it and you put it in artificial intelligence. So artificial intelligence is just a baby without any physical body. There's a brain. So what we do is we implement these 4 characteristics for that baby, so virtual baby, and then we give it data. Like you have eyes, you will see all those things. Your ChatGPT will see your questions and give you answers. Yeah. Similarly, we'll just give all these 4 personalities. Okay. And to evaluate the water quality. The heading was water quality. You guys remember, right? You wrote it down.
Yes, sir.
I can see half people saying yes. Write it down. So what's the need to evaluate water quality? Why do we need it?
whether it is clean or
Because it is pure or not pure.
Check pollution level. To protect the ecosystem also.
bacteria.
And to maintain the recreational activities. Okay? Yes, that is pollution level. So we can use artificial intelligence to what? Water quality assessment. Okay? Just read this out loud. Rhythm. Once. Very good. Now, will you guys say it or you want it also? are the 5 most important things you need. To do all this, we need again data. Right? Data is everything. Just saying, we need data for everything. How many of you are interested in computer science? Computer science. We are writing. Left hand with hands up and right hand. Yeah, very good. So for data is everything in computer science. The cyber security team is protecting the data. The software developer team is using the data. AI team is analyzing the data for future predictions. Everything is related to data. Okay. So for water quality, what all data do we need? There are 3 parameters. You will have all the videos. It's okay. I'll show you the links. Physical parameters, chemical parameters, biological parameters. Okay. Let me ask someone who will say physical parameters. Stand up and say. Transparency. Chemical parameters. So these are the 3 parameters to quantify any water quality. So what you will do? Remember the 4 quantities, the 4 qualities of quality. Yeah. So first is reading. You will read to quantify the data. That is yours. Second is collecting water parameters and values with ear of any water body. Okay. What is this? This is your learning. You are learning for your AI. You will develop it. So this is your learning part. Reasoning. Identifying the main issues with that water body. Why is it happening? What's the main issue? If there is sulfate, why is it sulfate? Nitrates, agricultural waste. And then what will come the mitigation solutions? What is this?
Question solutions.
What can you do? So what's
It's a camera device.
yes. And turning into data. That is your work again. So creativity was the last part. You will have to do creative work on your own. Let me give an example for this. Your Varthur lake. There was too much data collected on it, and what was the issue? Yes. Sewage water was the issue, right? And what was the mitigation solution? Treatment plant, yeah. It is not building walls against the lake. So this is to classify your emissions. When you quantify all the data, so there are classes. The water quality index, like similar to air quality index you heard. If the air is good, bad, excellent, very poor, like Delhi and Bangalore, you will see the difference. So in that category, there is water quality institute, water quality index. So there are classes A, B, C, D, E. Any anyone will say, hey, behind you. Drinking water for conventional drinking for wet. It's okay, it's okay. It's your hand. Water for agriculture and water for irrigation and industry. So we collected all the data like pH, connectivity, BOD. So when you get the data, you categorize them where it lies. So if the pH is 8, DO is 9, BOD is 3, and it comes in water for outdoor purposes only. So with this, when you take an AI, I'll explain how to do that. What that AI does, you take all your data, it will make it into database classes, what pH level classes, how many data is there, and feed it into the AI algorithm. So whenever a new person asks that AI, I have a lake with this much pH, this much pH, this much COD, DO, everything, all the parameters, it can tell what sort of mitigation solutions can be used or how, which class does it lie in, everything. You have to make a model on your own. And this, that will give out of this prediction. So building the AI model in your system, how do we do it? There are three parts. First is framework, second is the NPN, natural language processing. Chatbot is your normal, we have used it, ChatGPT and all. This is to show the data. The AI framework is when you're making a quality, your B, that's the main part, right? Yeah, that is your base. You create everything, that's the application, that's the framework. AI model is your wheel rotating in your quadri, the working method. How do you use that? And lastly, the chatbot. Okay. AI framework. I've given the most popular framework. You can write it down when you are making it. It's okay. So, what's framework? Don't read it. If you want, you can write it quietly. That's okay. The frameworks provide a foundation. Like I said, this is your base application where you will add all your algorithms, all your data, and make the chatbots, everything. This is the base. These two are written down?
Yes, sir.
Yes?
Yes, sir.
Still waiting for yes.
Yes, sir.
Then natural language processing and language models. This is your algorithm plus data. So with natural language processing, you can combine your human language with your data and everything. You can, how do you want to quantify it, all those algorithms. You can do that by using two models. There's Fast API, you can download it on GitHub, it is openly available. And there's Hugging Free Transformers. I don't know why they named it, but it is very useful. Okay? We will have videos, we will have this PDF.
Yes, sir.
Again, chatbot. The chatbot is just for the communication part. When you have the data, you want to communicate with the user, right? Yes. For that, we need an intelligent information system which can quantify the data and say it in a human way. It shouldn't say just pH this, DOD this, class this. So you should say that yes, your pH is in a human language. Okay. So there are chatbot tools available open source. There is Open Assistant and FoxPoint. Okay. You understood all the three parts, right? This will be all you have to search. Then again, GUI part. You can't just say that pH is this or BOD is this. You have to make it interactive for the user. Colors. Whatever website you visit, is it just boxes? No, there's colors, it's images, there's user-friendly interface, right? Yeah, you have to make that. You can make that in your smartphone also, laptop also. You know, app building, website making, HTML. Yes. In HTML, you must have seen, writing it for the user. Websites. You have to make, how to do colors in HTML also. Yes. Do it properly. That's it. Don't make orange and violet. And lastly, document everything you do. That's the procedure we have done. So that if you're doing it in a group, let's just say, one by one. First point. Yes. And anyone, what do you mean by all this? And. Whenever you are doing a group project, this is very useful. You will have direct information of what the other person is using, what all you have done mistakes. So you will be prepared to documentation, and this is for the report also you will generate for the Lake Symposium for your project. Okay? Any questions? All understood?
Then water pollution and pollution. Why our fish is more important than other organism or something? Why? Because fishes are good indicators of the water quality and ecosystem. Then water quality, how we can understand? Then we talk about that now. The different parameters, physical parameters. In that way, we can understand the fish. Then, because of how the fish, because of fish, how we can understand the water quality, water is good or not. Because fish that's happen in our lake or something, then that water is good or bad. Bad. That means our fishes are more sensitive. Okay, they are. And second point, they live under water and depends on dissolved oxygen, shelter, food, etc. Okay, so they cannot move from one place to other place because they want water. The birds can fly from other place to other place. Then our animals can move from one place to another place. But this fish, without water, they can move. Okay, so water is very important for fish. We want also water now. Then fish is nutritious. Yes, okay, that's why we can use fish as a food. Then demand for freshwater. We have two types of fishes: freshwater fishes and marine fishes. In that freshwater fishes, earlier people are more using the marine fishes. Now freshwater fishes are very important. Why? Because people realize that the health and the fitness value of eating. So the general concept on fish, okay, that the fish study, that ethology, that came from the Greek. That it is, or it is, that means fish, Then constricts this, constricts means cartilage and fix this means fish. So that main thing from the cartilaginous fish. Then osteitis, osti means bone. That's why bony fish. These are our bony fish. Can you tell some name? Bony fish. In our aquatic food chain, okay, it based on our fish, we have three different types of fish. One is piscivorous fish, then planktivorous fish, and vermivorous fish. The vermivorous fish we can found they are in the bottom of water. Okay, so in the bottom, what they will eat means they will eat the bottom. What it will be there? Degraded, the organic matters, then degraded macrophytes, and all the things. They will go and eat that. Then automatically they will disturb the sediments. For this sediments, that is the nutrient release into the water column. So our producer, primary producer is the microalgae or phytoplankton. They will use these nutrients and they will make their own food and they will grow very well. So our zooplankton, what it will do? That is zooplankton will eat microalgae. Then planktivorous, that name itself that what they will eat, we can understand. They will eat planktons, planktons like zooplankton and phytoplankton. Then piscivorous, that name itself, pisci means fish. They will eat fish like small planktivorous fish. Now you understood? Yes, ma'am. So based on the that fishing this one, they have surface feeder, they are column feeder and bottom water. That is based on the mouth also, it's different. Okay, the surface feeder, they have the uptake mouth. Okay, for example, the cutla, you can see the mouth. Okay, the column feeder, there is the straight mouth. For example, our that is the thing. Then the common cargo, that is our bottom feeder, the mouth round tail. So they are a, we can understand they are a bottom feeder. Okay. Then for your study, we wanted to know the how we can identify the fishes. We wanted to know the identification. So the fish has mouth, then eye, then different types of fins also is there. Caudal fin, then caudal fin, dorsal fin, then anal fin, then pelvic fin and the pectoral fin. Then these fins, why they fins they will use for the swimming. They want to move from one place to other place. Then this the fins, the dorsal fin has two types. The softer layer also is the spines. Spines means they will use this for the defense purpose. Okay. Then the scientific classification that our students are shown the birds classification, and that we have to do the scientific classification based on our particular the species, the fish species. Okay. Then in our some example I can give here the fishes in our bamboo lake. Here the fish species we have 12 different types of fish species. Then the common name, then the Kannada name also. That is Katla, that is Kannada is Katla only. Then Oreoprom is Eroticus and Mozambikus. We will call it as tilapia. Here the Kannada. Okay. You like this? Yes. Okay. These are the sound fish food. We can get it from fish. Then for your project work, you can use these are the some links that Indian biodiversity portal, then Animalia, then fish race, then IISC, that is our website, then the IUCN red list, that they will show the status, different status. all the things needed for this things. Okay, this is our Bangalore. Here, our Bharat sir and the madam is showed the thing. Then here we have the KC means Koranagara and the Chalakata Valley. Then H means means Nagara Valley and the V means Shabavati Valley. Okay, here your Varthur lake is here. Okay, then when you will go for your field work and all the things, before that is the madam shows how to identify your location and all the things with the mobile phone. Okay, then there is some example. This is the KC Valley lake, Bangalore, your lake, Kalandur lake, also Lake Kolara, and Varthur. These are the some examples of the lakes. Then we have the status of lakes that like the sewage water entering to the lakes, then solid waste dumping, then macrophyte power, then lake of no face. Lake of no face, then what happened? Then you can easily throw the waste and everything. Then the constructions activity near to the lakes, then building the bridges near to the lakes, then algal bloom, higher nutrients, then algal bloom can happen. Then fish, there you can see lot of fishes in our also lake. This happen here. Then you all are know the foam, Varthur foam and Kalandur foam. Yeah, that also. Okay, so our the environmental factors affecting fish water. That means our water quality parameters is very important to our study. Here, abiotic and biotic parameters are there. Here, the biotic like the prey-predator relationship and algal diversity, then other microorganisms. The abiotic factors, our Ratana madam will talk about that, temperature, pH, then TVRAMACHANDRA and all the things. This is our fish. So, for example, I will give one about Varthur study. Our sir and madam all are did in the 2011. Here, the 1962, we have some three species like Anis, Bathachas, Heteromustus, Fossili and Monsters species. Then 1998, there some our major cups like Indian major cups like Catla, Napi, Lotina, all the things are presented. Then we introduced Cladius garipinus also. The Cladius garipinus have the air breathing organs. That means they can take air from the atmosphere. Okay, they will all the fishes where they will take the oxygen water. But this species has two types of ability. That means they can take oxygen from the water and they can take oxygen from the atmosphere. So they will survive very fast. Okay. Then another thing they are a carnivorous fish. Carnivorous fish means they will then 2009 you can see that already that fish is there. Okay, that this is the species, the Cladius garipinus. You see this friend? Catfish, yeah. Then 2016, very surprise. That also disappeared. You can see here. Yeah. You can see they can take oxygen from water and oxygen from the atmosphere. Then also they are not surviving means what is the situation about that Varthur and Kalandur lake water? That means polluted. So if the catfish cannot survive in Varthur lake water, no other species can. Okay. Likewise, the water quality is very important. Then fish mortality in lakes of Bangalore. This is 2016. In our also lake, this is happened. Here, this is the some example I am just given to you. Here we can see that the different lakes that the fish that it happened in the different rivers also. So here, the everything, most of the things because of the sewage pesticides and other anthropogenic activities. Another thing, do not harvest fish during this spawning period. That is like mainly that is June to September time. That is we never harvest the fishes because the timing the small fishes are there. So we never harvest the fish during this spawning period. Then do not harvest juveniles. Small fishes also we never harvest. Then do not allow the introduction of exotic species in the lakes. Then educate locals about the importance and the ecology of freshwater fishes. So, your successful project, what you wanted to do? You wanted to select your project. Then prepare a project. Like what all things you wanted to do means you can find out the methodology and method part. Then what subject you are more interested, that only you can select. Then field visit and collect maximum data. Okay, for fish study you will then fish wherever the fishing activity is happening, that particular day you wanted to go and collect the data. Then analysis of data, like water quality analysis or other type of analysis you can do. Then result interpretation and report making. Then presentation, that is very important. In our late symposium you can present your project. Then successful, that will become a successful project. Okay, for fish diagnosis you can do the fish identification, then list out all the species in the lake, then find out the scientific classification, then food and feeding habit of fish, then find out the habitat quality like water quality and all, then list out their economic importance and industrial status, then so save lakes for fish, do not pollute water bodies. This is your food rate, and this fish are singing water pollution, then solid waste dumping and all the form of fish. Okay, thank you.
So they are tiny organisms moving in the water with the help of water currents. Okay. So today I will be talking about microalgae or phytoplankton. Then zooplankton. So do you know about the footweb? So in the footweb, since I showed you, there are primary producers. So primary producers are algae or macrophyte. They are green in color and they have the ability to photosynthesize. Do you know what is photosynthesis? Yes, ma'am. So since the algae are green in color, they have the chlorophyll and they photosynthesize, they make their own food. So then that food is transferred to the next organism, which is zooplankton, fishes, etc. Okay. So algae are the primary producers. Then next is consumers. So who will consume algae? Primary consumer, that is fish. Primary consumers are zooplanktons. Okay, so fishes are eaten by zooplanktons. So here you can see now. So this is the primary producer algae. Okay, so these are zooplanktons. Then these zooplanktons will be eaten by small fishes, that is secondary consumer. Then that small fish will be eaten by the bigger fishes and birds. The other tertiary consumers. Okay. And who are the decomposers? Bacteria, yeah, sure. So bacteria are the decomposers. Yeah. All the bacteria, they will decompose all the organisms after their decomposition. So planktons, they are organisms that move freely in water. Okay. So there are two types. One is phytoplankton, another is zooplankton. So phyto means what? Plants. Yeah. So phyto means plants. So phytoplankton means plant. They are organisms like plants.
stones, then tree barks, etc. Okay. So what are the general characteristics of algae? They have the ability to photosynthesis. So here you can see that carbon dioxide is got in the presence of sunlight. The carbohydrates are made. Then with the release of oxygen. So earlier our atmosphere was reducing, but it is because of algae activity about 3.5 billion years ago we got the oxygen. So we have to thank algae because we got oxygen because of algae. Okay, or else we won't be able to survive. Next is like algae of two types. Do you understand prokaryotes and eukaryotes? What do you mean by prokaryotes? Okay. Yeah. Prokaryotes means they don't have proper cells. But eukaryotes have proper cell wall. So only cyanobacteria is a prokaryotic organism in the phytoplankton. All the other groups all belongs to eukaryotes. Okay, so cyanobacteria is only a prokaryote. So algae, they can be microscopic or macroscopic, they can be unicellular organism or multicellular organism. So this is a plankton net. Okay, so according to Ramachandra 2008, so about 50 liters has to be poured through this plankton net. Okay, so here at the bottom you can see it? This is small tube. Okay, so when we pour water through this, all the water will be poured out, and only the algae, our zooplankton, will be accumulated at the bottom of this tube. Then we have to transfer this to a sterile container, then preserve it with 4% formalin. Then we have to take to the laboratory and look under this microscope. So algae looks like this, okay, under microscope. Okay. So why should we do a plankton analysis? Actually, it helps in understanding the quality of water. So we all do some chemical experiments in our time. So with that chemical experiment, sometimes we won't be able to completely define water quality. So with the help of this plankton analysis, we can understand whether water is polluted or not. So in polluted water, only organisms which is able to survive in polluted water, they only will survive in that water. All the other organisms, sensitive organisms, they will die out. Okay. So we can understand the pollution content of that water body. So this is some type of algae. So there are 4 types: basidiophytes. So here you can see these are basidiophytes. They are brown to greenish in color, and so that's how mainly we can understand this basidiophyte. Okay. The euglenophita, they have, here you can see, they have flagella, and also there will be a red eye spot here. So if you see that, you can understand that they are euglenophores. Then next is cyanobacteria. Cyanobacteria are typically blue-green in color. So when you look under the microscope, if you feel that the color is blue-green, then definitely it belongs to cyanobacteria. Then chlorophyte, they are the green algae. They are typically green in color. Okay. So this is cyanobacteria, and these are chlorophyte here. So this is difference. Like you can see here that clean up. This is clean water algae. This is polluted water algae. Okay. So these looks different, isn't it?
Yeah.
Yes, ma'am. So only some algae which is able to survive only in clean water, they can survive there. They cannot survive in polluted water at all. Okay. So guys, at some like euglena, they are found in only organically polluted water, because there will be less dissolved oxygen content, and those higher amount of urea and CO2. Okay. So next is zooplankton. They are animal-like organisms that move in the water with the water currents, and they are food to fishes. Okay. And this zooplankton eat algae, bacteria, protozoa, and other organisms. So this is how zooplankton looks like under the microscope. Okay. So with the help of microscope only you can see phytoplankton and zooplankton. So these are the types: protozoa, orthopod, parasite, copepod. So have you seen? Do you know this? Paramecium and amoeba. These comes under protozoa. So they are basically present in very much polluted waters. So if you see and look under the microscope, then you can see their numbers more high. This amoeba, paramecium, they are high in number. You can understand the it looks like, and they are carnivorous in nature, and they feed on other zooplankton, so they can eat all the other zooplanktons and alligator. Okay. So, what is the causes of water pollution? What are the causes of water pollution? Okay, thank you. So this section you can see that livestock waste and pesticides, all the waste, then pollution with car wash and then household waste, everything, if it ends up in water body, all the organisms will be affected. So because of this pollution, all the ionic content, the nutrients, everything will increase, nitrate, phosphate, and all the ions, all will increase. Okay. So if water is polluted, then all the organisms in the food chain. I told you, primary producer, consumers, and decomposers, everybody will be affected. So if water is polluted, then what is the condition? Higher diversity, lower diversity. Okay. So if water is polluted, then there will be lesser fishing activity, isn't it? The fish that will be happening, so there will be less fishes. Then, so there will be overall what will happen? There will be overall reduction in the cash price, isn't it? Then lot of what to say? That KKEHS showed in Varthur. So in Varthur, because of pollution, there was a lot of mosquitoes. Mosquito breeding was there. So a lot of cholera, all the diseases had come to all the malaria, everything has come to all the images around it. So likewise, there will be some medical cost will also be affected, and overall there will be a very much loss in the biodiversity also. Okay. So these are some databases which you can refer to for algae, zooplankton, then macrophytes, etc. Okay. So what you can do if you are going to a lake, you can do all these projects, like you can monitor water quality, then you can look at the microalgae diversity, then zooplankton diversity, then fish diversity, then bird diversity. Okay. Then you can find out all the ecosystem services provided by algae, zooplankton, birds, fishes, etc. Then also you can work on the bioremediation process. That is the noodle project. I think that will be a little more at your level you can do all these projects. Okay. So same makes sense biodiversity, so the pollution. So ensure clean water, air, and soil for healthy. Thank you.
Okay, we start with water quality sampling and analysis. So, tell me what are the importance of water quality? Now, what is water pollution? Okay, when do we say the water is polluted? Those are all the collective things. The basic thing is, see, the water needs a certain amount of nutrients, elements, or ionic elements, for basic amount. The fishes need it, no? For it to survive. When the threshold level is exceeded, we need a certain amount of sugar in our body. When it exceeds, what happens? You get diabetes. The same way when the pollution, the also told Varthur Lake, you saw the dropping happening. Yes, ma'am. Why does that happen? Because of the calcium phosphate. Nutrient, basically nutrient. You can see the excess amount of growth of macrophytes. That is also due to increase in nutrient content. Okay. So why do we need to know water quality analysis? Whether to know the water is polluted or not. What is the second thing? How it is polluted. The third thing is what is the specific source that is affected. So water sampling, what is it? We collect the sample, we do the analysis and we see whether it is polluted or not. How do we see whether it is polluted or not? We compare it with the water quality standards that is given by the main board. European standards or other standards are there. We compare it to our water quality values, whatever they are called. We compare it with the other standards and say, okay, the water that I did the analysis for us to work on. So when you go for field visit, what are the things you need? Okay, you'll be reading all these things. Since there is no time, you guys, you can get all this in the website. So coming to the main point, you visit the Varthur Lake. So Tulika ma'am showed the map of Varthur Lake. She showed the inlet point where you can select the inlet GPS point. Then you can map your study area. So for the water collection, water sample collection, we take the first point at inlet. Since you have showed how the water, wastewater enters the water body. What are the sources ma'am showed? Sewage, industrial effluents, high, low, okay, sewage, and when we have precipitation, the rainwater runoff, what will we collect? All the dust from the urban area will all enter the water body. So that is the inlet. That is where the pollutants are entering the water body. So we take sample at the inlet. That is the first sample collection point. Second point, where we take at the center. So since you guys can't reach the center of the lake, you can take either here or at this spot and say that is the center. Why do we take the center? Because the center region will give us the overall general water quality. And the third point is at the outlet. So why do we take at the outlet? These lakes, they help in reducing the pollution content. After the water is stored for some days in the lake, they help in purifying the water a little. So to know how much, what is the water quality at the outlet, we take one sample point collection at the outlet. Okay, what are the 3 points? So they all showed, Abhishek sir showed, and since Yasmin both of them showed what are the physical, chemical and biological parameters, we go to the next step. So these are the standard methods that we follow. The parameters that divide into on-site parameters and off-site parameters. Why do we do that? We test the on-site parameters such as water temperature, air temperature, pH, electrical conductivity, and pH at the on-site. On-site is when you collect the water sample, you will do all these 5 parameter analysis there itself. Because all these parameters are affected by like outside temperature, the pH changes by the time you bring it to the lab and do the analysis. The pH would have changed, or due to your water reacting with the atmospheric air, that pH would have increased or reduced. So to avoid all these things, we do all these parameter analysis on site. Okay, what are the on-site parameters? Off-site parameters are all these things. There are even more parameters, but today we will be seeing only these parameters. Okay? The first parameter is temperature. How do we measure temperature? With this instrument. What is this? When you analyze, when you want to know the air temperature, first you will take down the air temperature. To check the air temperature, you need to hold it. Make sure you are not standing, directly under the sun, because this picks up the temperature very quickly. Mercury temperature. And also you should not hold it with your hands. The mercury tip that is there. Why? Because it picks up your body temperature. So make sure when you are at the side, you are in a little bit of a shaded area, and then you take the air temperature. For analyzing the water temperature, you can see here they have inserted the thermometer in the glass beaker. You take the sample. This is the Tarzan cup. You fill it up, and you insert the thermometer in it. Wait for some time. When the mercury level reaches a stable line, then you take the temperature value. What is the unit for temperature?
Degree
Celsius. Degree Celsius. That is what I want. Guess what is pH?
surface of the
The pH is the nothing but the H's around concentration. How do we measure it? Like what does it show? Whether the water is alkaline or acidic or basic. So what is the pH meter range? What is the scale of pH? 0 to 14. What is when do we say the water is acidic? When it is 0
0 to 14.
7. When it is from 0 to 7, we say the water is acidic. When it is from 7 to 14, we say the water is basic. When it is exactly 7, we say the water is neutral. But for water, we take it as 6.5 to 8.5 is the minimum pH. That is the neutral pH that the water should have. So this is the pH meter flow. So there is a on and off button here. That is the pH meter. There is a on and off button here. You on it, you switch it on. You will be having the same water sample that you use for temperature analysis. After oning the probe, you immerse it in the water sample. Wait till the value stabilizes, and then you take down the reading. Okay? Is there any unit? As a unit for temperature? Sorry, pH. What is the unit for pH? No, there is no unit for pH. We just write the value. Okay? Then after removing the probe outside the water sample, you need to make sure you wash it with the distilled water. Rinse it with distilled water, so that when you go for the next analysis, it is not calibrated with the value doesn't take up the last reading. And you use the tissue paper to grab and dry the electrode instead. So the next one is total dissolved solids. What are total dissolved solids? That include the organic pollutants that are there, organic matters, inorganic and mainly minerals. All this combined together, and we say what is the total dissolved solids that are there. How do we estimate? So, higher concentration of total dissolved solids, what will they do? They, the water will be very turbid. You can't see that the water is clear. There won't be clear water, very muddy water, or very, when the sewage is entering the water will be very green, grey color water. Black color water. So how do that happen? When there is too much of pollutants that are entered, that will increase the total dissolved solids content. And then water temperature will increase. How will that happen? These dissolved solids that are there now, they capture the temperature. They hold the temperature and increase the water temperature, overall water temperature. That happens. And next is decrease in photosynthesis. For photosynthesis, what is the main thing that they need? Sunlight. When this pH level increases, the water becomes too turbid. They won't need sunlight entering the water body. So the photosynthesis rate will reduce. How do we estimate TDS? This is the probe for TDS estimation. There is a on button. You click on it. It will give a Then you take the reading. For TDS, the SI unit is parts per million, ppm. Okay? And then the same probe will give the water temperature also. You will note down the water temperature value and TDS value. Okay? What is the unit of TDS?
ppm.
Now after removing it, what do we need to do?
Wash it and
Yes, very good. Next. Next is electrical conductivity. Why is it measured? Because it gives the what is the conductance of the water. So this gives TDS gives the overall organic matter that is there, inorganic and metals. Electrical conductivity will mainly give the inorganic impurities, the inorganic content that is there. So water to conduct electricity, it needs to have ions. No? So the more the ions, the more EC value will be there. So how do we do the same probe that was there for TDS? It's a similar probe. We on the probe, immerse it in the water sample, get the wait for the value to stabilize, take down the reading. EC value will be given in microsiemens per centimeter. Okay? All this, the unit, everything will be there in the website. You guys can note it down. So it will be given in microsiemens per centimeter. Then you after noting down, you will remove the thing, remove the probe, still got and you will mix it and tap it with the machine. So what is dissolved oxygen? Do we require dissolved oxygen?
Yes, ma'am.
Why do we? As we take oxygen like in the air, fishes take oxygen water. That is the dissolved oxygen that should be required. So the minimum amount of dissolved oxygen that should be there, minimum is like that must be the number. That is 4 mg per liter. The minimum dissolved oxygen amount is 4 mg per liter. If the water is not having that much of dissolved oxygen, if it is like 0, 1 or 2, yes, that is when we see the excess amount of fish death in lake water bodies or any rivers. Okay. So how do we do the analysis for DO? This is the BOD bottle. Okay. So when you are taking the after you take the sample in your sample water bottles or you collect it in a bucket, you can't access directly from the sample bottle. Then you take the BOD bottle. You need to go this way, add water very slowly so that there is no bubbling that happens. Why? Can you see here the oxygen, the main source is from diffusion of oxygen from air and also photosynthesis. During photosynthesis, CO2 is consumed and oxygen is released. So these two are the main things that are the source for dissolved oxygen in water. So when you are adding the water, now make sure there is no air bubbles that are formed. You need to add very slowly near the walls of the BOD bottle. Okay? After you add, you close the lid and tilt it upside down. The excess of water that will be there, that will fall out, and this will be your DO. The water you need for DO estimation. After you do that, we will take we take 1 ml of MnSO4 and I am adding 1 ml of air. And can you see this brown color dot that is formed? This shows that there is the presence of DO. If the water is too polluted, the flock will be white, copper fully white. So that shows there is 0 amount of DO that is there in the water. Then you mix it properly and you have to keep it in dark place for 10 minutes. Now there is no dark place, I am keeping it here. We will continue with that estimation later. Now I will go to DO. So what is, now these are all offset parameters. What is BOD?
Biological
When you use that water, fully, you fill these two bottles fully with just the aerated water. Okay? Understood? And in the other two bottles, you will add 5 ml of your sample. How much? 5 ml. Yes. And the remaining with the aerated water that you have got. Okay? And in that, you will name it as day 1, day 5. Here also, what do you name it as? Day 1 and day 5. Whatever the day 5 bottles are there, you will keep it in a dark place. Okay? For 5 days. You won't touch it. We will do the pH estimation on the 5th day to know how much of oxygen those microbes that will be there in the DO water will consume. On the first day, we will do the DO estimation that we showed. I added evidence of for care. That I will do it. That will be doing it on the first day. So next is total hardness. So you know what is total hardness? It is mainly because of calcium and magnesium that are there. You know, when the water is very hard, how much ever soap or shampoo you add, the water doesn't get that target. That won't happen. You need to add more detergent to make the water very target. Did you get it? Yes. So that is because of either temporary hardness or permanent hardness. If the water is temporarily hard, how do we remove the hardness? By boiling the water. See, when you boil the water, can you see the white salts that get settled in the water? Yeah, that is because of temporary hardness. The whatever the bicarbonates and carbonates that are there, when we boil it, they get settled. That is how we remove the temporary hardness. For permanent hardness, that is because of chlorides and sulfates. They remove it with the different instruments. So next thing is this. Okay, for estimating the total hardness, you will take 25 ml of water sample. This is the measuring cylinder. You will take your sample 25 ml in a conical glass. This is a conical glass. You take 25 ml. Then you add 1 to 2 ml of water solution, and then you add a pinch of video black tea indicator. So that will give you a pink color. Can you all see the pink color? Yes. Then you titrate. Then you do the titration again with EDTA. EDTA is diethylamine tetracyclic acid. When you do the titration, you get this blue color. That will be your endpoint. Whatever the titration you do, you all have used unit stand. Unit. Okay. See, can you see this unit? This is the unit, and to hold that unit, there will be a stand. That is the thing you use. In that unit, you will be adding EDTA and do the titration. So when you get that blue, that is the endpoint. You will note down the reading. Okay? And you do this titration for 3 times to make sure you get the concordant value. Concordant is saying you need to get the repeated value, the same value, the endpoint. Okay, then you note down the reading. This formula is you will insert the concordant value that you have got in here, the ml of EDTA used into 1000 divided by how much of sample we do? 25. That value you will do and get the hardness. So calcium hardness, this is the one of the things that is, causes the total hardness. Main thing that causes the total hardness. How do we estimate? You take the same 25 ml of sample, we add a pinch of peroxide indicator. Then with the same EDTA, we do the titration. When the color changes from, can you see pink to what is it changing? That will be your endpoint. You note down the value, you do the titration for 3 more times, get the concordant value and put it in the you will put your value in this formula and get the, that will be finally, magnesium harness. To do this estimation, it's very easy. We don't have to do any analysis. This is the formula. Magnesium harness is equal to total hardness minus calcium harness. You would have gotten the total hardness value, calcium harness value. You subtract it, then you will get the magnesium harness. See, usually your total for the total hardness after estimating calcium and magnesium hardness, usually for all the water bodies, calcium hardness will be high. If your magnesium hardness is high, that will be because of the increase in the amount of nutrients that are there in the water. Okay, this you need to remember. So for alkalinity, you know, this is the water is trying to neutralize the studies the pH. No? Water will be, if it is below 7, it is acidic, or above 7, it is basic. What will the water do is it will make sure to keep the whole water body in the neutralized condition. To make it not be very acidic. Do you understand? To make sure the water doesn't become acidic, it will try to keep the water in neutral condition. To estimate that, it will do the alkalinity. Next is the chlorides. So this is nothing but the salt content will be there in water. To estimate what is the chloride concentration, we do the chloride estimation. If the chloride is in high content, that is because of the waste waters that are entering in the water, which will be containing high amount of chloride. This is the formula that is there. So to estimate this, you will take 25 ml of sample, add 2 to 3 drops of potassium chlorate. You will get this yellow color, and then you do the titration with silver nitrate. So you will be using silver nitrate is the costliest chemical. So you will be using the costliest chemical to estimate the chlorides, and your endpoint will be yellow to that color. Okay. So after getting the concordant value, I showed you the formula. You will be adding the value in that formula and getting the chloride value. So finally, we do all this analysis. What do we get? We have the values. Can we ignore all the values after we get? These are the inland surface water standards that are given by Bureau of Indian Standards or CPC. CPC, Central Pollution Control Board. They have given these standards. Say I get pH of 8.7. That is not in this range. Then I say my water is polluted. The same way I check for all the single parameters that are there. See in which class my water quality belongs to. If it is meeting all these values, all the values that I have got are within this range, then I say the water is suitable for drinking water. If it is in class D, I say it is used for outdoor drinking. The same way they have given different classes. This is for surface water, like lake water bombs. This is for drinking water. You all have, do you use RO water? Aquaguard, those waters. So you can test those waters also. Make sure they have a minimum of these values. Because what those aquaguards do, they filter too much of minerals that we need. They may have a very minute amount of minerals. So you can do your, for the lake symposium, you can take different larger waters, do the analysis, and say whether all those RO waters are having a minimum of these things. Okay? Then the same with you are seeing in the morning session. This is the website that is there. When you go click on the reference materials, you can see environmental monitoring. Then it will take you to this page, to this section 7 is there. When you click on it, you will get all the principles, what is the procedure that needs to be followed to each parameter, and how to prepare the reagents. Everything, the formula, everything is given in that. Now I close the lid, take down the excess of water that is there and can you see all the flocculants were dissolved? You wait till everything gets dissolved, you will be doing this till then. Now you will take of the sample that was there in that bottle. And you add 2 to 3 drops of starch. This is the so this will be dark blue. Okay. I have taken sodium peroxide in this bed, not a different application. So you add it drop by drop. Can you see this now? You can see the little dark blue. Now I have to hold it very slowly. Again, purple. Right now, I add one more drop. This is your endpoint. This is where you check. Now it is transparent. And now you hold the thing. You see what is the reading is there. How much the value is there, you note down. You do the same thing for 3 times. Concordant values, you should get the same value 3 times. Make sure you are getting the endpoint at the same value. That value you take down. Whatever the formula that was there now, you will put this value there and then you do the calculation. Okay? Understood? Yes.
So with all the topics out. So now what you all will do? All the topics you have gathered, the information has come into date. Now you all will compete with each other to come into the next symposium to get an award. Right? So there should be some healthy competition. To be cannot do all sides should be there. Right? Likewise, in some competitive world or in this competitive world, some of the species are there. These are called macrophytes. What are called? Macrophytes. What is macrophytes? Macro means big and phyte means land. So in this universe, most of the world, W-O-R-D, not the okay? But what you will say that is derived from the Greek word. Most of the word, what you will say, okay? Greek word or Latin word. So from this macro, that means big, and Python means land. Okay? So this is how it has been said. So this macrophytes are classified or categorized into three forms. Some are submerged. Submerged is in the water. So these macrophytes, basically are found where the water bodies are living. And these are also called bioindicators. Bio means life, and indicator means indicator. That means it indicates the life. So where the life will be there, as you all know about water, what you drink, that is called life. Basically, you all say life. So where these species reside, they give the indication of some life. So that is why they are also called bioindicators. So as I said, these are classified into three. Okay, three what are there? Some species are submerged. Submerged means they are fully in the water. Okay, some will be immersing. That means immersed. You can do. Okay, and some will be the floating. That will be float on the water. So these are classified into three or categorized into three. Okay. So this is the first ecology. Okay, how you identify any species? So on the base of the leaf, flowers, roots, flowering, or the leaves. What are the? So these are the basically categorized. How many species is identified? So for any water body, how you can identify any species? Okay. So first is Iconia. This is also called water hyacinth. So leaves that is 30-40 centimeter will be there. Then and it will be the spotty petiole. You know what is petiole? Petiole means here the leaf which is there, the small tip will be there. This is the plant, right? It is yellow. So this leaf which will be connected to this, okay, some will be thin. That is called petiole. That will be that big leaf. That will be giving some support. Okay, and that will be in the spongy. So that is iconic. And that will be in flowers. How you can identify the base of flowers? So 60 to 100 will be there, and it will be 6 petals, pink or purple. The color will be the pink or color. Purple. And each will be fibers or feeder lines. Feeder means bird feeders. So roots it will be like feeder. The feeder of any bird. So that is how you can identify any species like Iconia. Then the flowering it starts in October or it continues in September. And what are the uses of this Iconia? That can be used in the fodder, that is the pattern, it can be given food, and several things like paper also it can be in the industry. These are well and also ornamental. Okay? Then as well as thematics. And these are also called feeder mosquito fawn. So it is circular, it will be there, it will be round and triangular shape. Okay, it will be the triangular shape between this, the leaf will be there, that will be in the triangular shape will be there. Okay, first. Triangular shape and this will all be feeder. And the leaf, if I say about a small and overlapping, in this the leaf will be there, that will be overlap. Okay, one over the other. And then the roots that will be done with 12 to 15 mm, and young roots will be fusiform. That will be in the fuse. That will fuse. Any root which is there also as well as that will be fused together. Okay, like these two boys are there, they are fused. So this is fused. Okay. So the reproductive season that is November to February. And what are the uses of this species? That is food for the animal, bird species, and it also contains high proteins, vitamins, and minerals. And these also mosquito control and form thick layer on water surfaces. So they also give a barrier to mosquito. You know, they give the larva and they chlorine. So it will be so few in the water, or the mosquito will not come and chlorine. Okay, so that also will support in this water system of the mosquito. Now, so these eggs will be oval side, oval, egg, that shape will be oval. Okay, and it will be 1 to 8 mm and 0.6 mm it will be there. It will be broad and in the light green. The root will be single and 1 to 2 centimeter long. Okay, and the flower will be, so it rarely flower. Okay, rare, not every type of some of the species they flower in the spring, some of the summer or some spring there. So these species rarely flower. Okay, then flowering later or pink there, and some of the uses food for the particles, ducks, fishes, and used for the cuts and wounds. So if any cuts will go or any wounds are there, so by taking this species we can apply on the cuts or wounds. Okay, so these are the uses of this species. Okay, so now storage area. Okay, so these leaves were used to each plant. Each plant will be having 2 leaves, and then the rosy green leaves. So the color of these leaves will be green. The more green it will be there. And the root will be in the bundles, and it will be long pointed leaf plant. So here you can see the roots which are well pointed and having leaf on them. Okay, so this is how you can identify spirulina when you are going through something or identifying any species. Then the flowers produce buds that detach and sit on the bottom of the pond. Okay, is it clear? In the flowering season, October to November. And what are the uses of this species? So these are the food for the ducks and some fishes, and food for cattle and pig. So these are used for the food products of the species. So now, Varthur. So if anyone asks you which is the smallest species, plant species in this universe, that is Varthur, the smallest. Yeah. So this is tiny, very tiny. It's not tiny, it is very tiny. And with no leaves, no orchids. Why? Because it is so tiny, it is not having leaves or things, at least form it will be there or no. It won't characteristic, it will not be having. It is so tiny. And then the roots that will become in or less in the diameter, a smallest plant in this universe. Then flowering season, that will be the late spring, and used for the ducks, bees, and it also control. Okay, larva of the mosquito. Yeah.
Where do we find that?
Sorry.
Where do we find them?
Where can this species? Any water bodies. Okay, ponds, lakes, which are there nearby, that this species you can find. As I say in the first, these species are found near the water bodies. Okay. So now is here. So these leaves, these are 30 cm or 17 cm long and fan shaped. Okay, these species we have this fan shaped structure. Okay, these are there. It will be like this. Should be there or not? Yes. Okay. So flowers cluster in leaf and axils, and the inflorescence in the shape of the plant. It is the inflorescence there that happens in the center of this flower. And the roots, which are physical roots hand and branch, and 50 to 80 centimeters, it will be in the shape. And each root covered within 5 years. So we can define the which are there of this species. Okay? And then the flowering always occurs in the odd region. And then uses the feeding for the pigs and ducks and that is anti-inflammatory, anti-inflammatory, and also some of the some problems here or dysentery. If any stomach problem there, they will be also used. So now it is serving line. So this having the mat form, large powder quantity, the liquid is the OH side, and 0.2 is the centimeter round will be there. So the root, this will be multiplied in form evenly. So this these are it will be 2. And in this, in the root, what happens of this species, the leaves which are there, they will be modified to make the roots. Okay? They do not have single root or something like that. The leaves only modified into roots. Here just count. In the body term, we cannot say fact. It is called count of management. Then coming on, we can use science. So the power will be located and that will be the cluster. So that will be so long. So that is not tiny or small. That will be long. And then the powder is made to absorb and what are the uses of this staple food. There are several food are there which are made from this leaf. There are several countries where they from this leaf. Several vegetables are there, rice are there that are made from this leaf. Because there are good uses, like are there, vitamins are there, proteins are there. So several nutrients this has. So that is why government are there that is in the food color, but in the green and not in the yellowish color. So the green is in the green flowers which are there that is in the green, yellow that is in the red. So this can be in the red, that is in the green. I said it start. Green is in the green and yellow is in the red. Okay. So now, like Puna, like Islam. So these are small kinds of students. And the of this power is white and purple. But that is together is a purple. That is how you can identify this continuum based on this power. Then will be very difficult or narrow shape. Narrow shape here for this species. And the state will be always be the greatest in the age. The state will be there that will be the rate of greatest value. And followed by the government state who can identify and can now November to March. And what are the uses of this species? That is for the angle and even for the angle and the okay. So the outcome of this the leaves which are there, which are made and if any person which has some physical problem, we can use. Then some mud plants. So that some mud plants which are there. What are some mud plants? What I am saying, some mud means to be so these will be flat line and pointed and wide and sharp. Use sharp to cut anything sharp to suppose this lake is your zero. This lake, so this lake like this, this look like this. The structure of this lake will be sharp. But anything are there. Likewise, the leaf of this will be then it will be more pointed. The state like that and form base side. This also make base matching water. And then cover quality that will be yellow, it will be there, white and silver on the surface. And this cover is the summer. And what are the uses of it? They are used in the manure. That is the carbon which are there. They use these species for the manure. Yeah. They try which are there. They try to they use the that is how white is used. White is called manure. Why manure is used? Right? shape. Some plays are there, so different explain the word. Different question, like what says that king and Naya. What Naya? Yes, Naya. Naya, I have said you earlier, most of the words have been derived from the Greek. So it's also Naya has been derived from the Greek word, and the Naya that is called Nayan word came out from Nayan. That means slow. Without giving up the noun, what is the meaning of pleasure? Yes, Naya. What is the lotus, like lotus, mapping of the subspecies material. They would have flowers in the water. And the flowers grow above the water surface, and they are found at a high temperature in diameter, and they have different range, like 2, 5, 2, these are the colors of different species, and 7 to 8 chickens are found. Chickens of the this flower.
Five petals.
What are the evils of this? Birds and seeds giving. The birds of this parents that will be giving. And the seeds of also giving. And these are having a property like antioxidant, anti-inflammatory, and anti-enzymes. So suppose if I say something, you will get answers. So if I will give some definition of this substance, that enzyme will be how all the group this is how we can and yeah, one more thing I would say, we want to say about this. One of the scientists he wished to deliver some lecture. After delivering his lecture, the man used to one of the students and asked that scientist that in your opinion, who is the scientist? That we are, in your opinion, is because that you as man was himself is scientist. So that we are, in your opinion, who is the scientist? You know what scientist is? He said he was this. He was this scientist. Because the chemistry together over years, and what you think and what you think, no one knows. You understand you. Okay? And you know who are the scientists? What you know the name? That one Dr. TVRAMACHANDRA.
So now we have come to the end of today's session, okay? So before leaving, Abhishek will show you the next web page. You can write down.
Anybody see it?
Write down.
Write it down. WGBIS .CES .IISC.ac.in. Over here we have all the information about our applications and databases. This is the resources, and over here we have links to the page. In this, we have all the important dates registration, and we have a session for students and teachers. Over here we will have your resource materials and the field conferences. Like today we had it in other schools also. Last was PGS. So you can see all the